Let's continue with the question 2.4. It's about genetics. We know that many students find challenges with genetics. Question 2.4 from National Senior Certificate 2022, NCS 2022. The diagram below shows the inheritance of flower color in snapdragon plants. The two alleles controlling the flower color are red and white. It means that because the two alleles are capital letter, this is a capital letter, and also this is a capital letter, it means that both of them are dominant. So if both of them are dominant, here it's showing us that you have a dominant red crossing with white, and it gives you pink. When you have red and you have white, we produce pink. So pink is neither red nor white, but is an intermediate between the two. So the type of dominance which is there, the moment you get an intermediate, neither of the parent color produced in the offspring, it means that that kind of dominance is called incomplete. It means that red is neither dominant over white and white is neither dominant over red. An example is different presidents of different countries. When they sit on one table, all of them, they will talk about issues affecting them equally. So it means that neither of the president of country A nor the president of country B is dominant over the other. Why? Both of them or all of them are presidents. So neither red nor white is dominant over the other. So when they combine, they produce an intermediate. So let's go to the questions and we see. They are saying that state the type of dominance shown by the snapdragon plants. So we have seen that it is incomplete dominance. If it was complete, then we would have got either red or white in the offsprings, or both red. Some of the organisms are red and some of them are white. That is complete. If it was codominance, we would have got a mixture of red and white. But now we get an intermediate, therefore it is incomplete dominance. Give a reason for your answer. The answer, we have explained it, that you produce an intermediate, or an intermediate color was produced. A gardener crossed two pink flowers. Use the genetic cross to show the ratio that is expected of when you cross two pink flowers. So it means that if both of them are dominant, definitely this is going to be capital R, capital W, capital W. Yes, they produce one gamete from this side and one gamete from that side. So this one is going to produce a capital R. This one is going to produce a capital W. So it means that this crosses with this, you produce this. And RW is an intermediate of red and white, which is pink. So it means that when you're talking about the genotype of pink, we have to use this. So what are you supposed to write? Okay. We are saying that now, the first thing you need to do is you say, because it is a second generation we are crossing, therefore it is advisable to use P2. So it's going to be P2. You tell us the P means parent. So P2, parent 2, because we have already parent 1. So now pink and pink, it means that that's P2. Yes, if it's the second generation, we use P2. P2, yes, we can say F2, second Fourier generation. We mark this as ones, like, so we give you one tick there. So don't forget, if you write P2 or P1, F1, it must be, yes, it must be P2, F2, P1, F1. If you write one, you don't write the other one, then you get it wrong. For example, P2 and don't write F2, you don't get a tick. So now P2, you tell us the phenotype. The phenotype is pink. We cross it with another pink flower. The X must be like this. It must not be like this. No, the X must be like this. Then you go to genotype. What's the genotype? We have seen the genotype here. That is capital W, capital R, or capital R, capital W. Therefore, we're going to have to say here, it is capital R, capital W, cross with capital R, capital W. Now, these must separate under meiosis. So you write the word meiosis. So meiosis, you form the gametes, capital R, capital W. Here, capital R, capital W. So we call these ones gametes. Gametes, don't leave these gametes naked. In most cases, I always tell my students not to leave these gametes naked. They're not at the beach. All right. We circle them. We circle them. Even if you don't circle them, still you get a tick. But I prefer to circle them. Why? So that we can differentiate them from the genotype. And always know that there must be a space between these two gametes. Don't combine them, because when you combine them, then it will look like it is a genotype. There must be spaces there. All right. Now these gametes, they're supposed to fertilize each other. Don't use the word fission. We won't give you a tick. Fertilization. When fertilization occurs, you can use a Punnett square, or you can use crosses. Since it is a monohybrid cross, you can use crosses. For example, this goes with that. So I'm going to have capital R, capital R, and now this with that. So it's going to be capital R, capital W. Now this with that is going to be capital R, capital W. Then this with that. So it's going to be capital W, capital W. Yes. So this is what called fertilization. So these are the offsprings of the parents. So F1 offsprings. What are they? They said snapdragon. It means that those are the offsprings or the snapdragons. But what was the question? The question is saying, use the genetic cross to show the ratio of the expected phenotype in the offspring. So we are looking for the ratio of offspring. Okay, let's look at the ratio of the offspring. Now, capital R, it is red. So I have red, How many are they? It's 1, comma. I have capital R, capital W. It is pink. Capital R, capital W is pink. So I have 2 pink. And then now I have capital W. It's white. I have 1 white. So basically I have 1, 2, 2, 2, 1. So that is it. So now when you are looking for a mark, when we mark, we are looking for ticks, and then sometimes we put what called a compulsory mark. So in case we come here, we do this, we do, yes, this and that, we give you a tick. Phenotype, we give you a tick, it must have a cross. Genotype, we give you a tick, it must have a cross here. If it doesn't have a cross, the tick might not be given to you. So we come to the gametes, we give you a tick, and then we have for crossing and you get the offsprings, we give you a tick. So because the question is saying give the ratio, so this one might be a compulsory mark. It might be a compulsory mark. So we give a tick, but we put the word C there, which means that it's compulsory. There is 1 mark short, which is meiosis and fertilization. Give you a tick. So how many marks are there? It's 1, 2, 3, 4, 5, 6, 7. And then in most cases, we give you 6 marks. And also sometimes cross, we can give you a tick there. So maybe we can say that this question is 6 marks. But how do we count? When you are counting, you first count this. However much you get all of them, like you have all of this. Without this now, for this case, you won't get 6. You have to be with all of them. You have to be with all of them plus this. Or you have to be with this plus another 5 so that we make them to be 6. So that's how we mark the genetic cross. This question, it is 2022 Department of Basic Education, NCS. Another question is about evolution. Here they're saying that diagram A and B show the ventral, that is the bottom side of the skull, skulls of 2 organisms. The diagrams are not drawn to scale. It means that they are not, that's not their size. So now asking, which diagram represents the skull of bipedal organism? Let me first explain the word skull. Some people, they confuse the word skull and cranium. Cranium is where the brain goes. The skull involves even the jaws. So when you are meaning where the brain goes, please don't use the word skull, use the word cranium. So skull is the whole thing, the head, including the jaws, the teeth, and everything. But when talk about cranium, please, we are talking about where the brain goes. All right. Now, which diagram represents the skull of bipedal? Bipedal, it means that organism is walking on 2 limbs. If the organism is walking on 2 limbs, it means that what shows we have 3 factors which you can identify whether the organism is walking on 2 limbs or not. The first one is the position of foramen and magnum. The second one is the shape of the spine, if it is S shape or C shape. S shape, it means that it allows flexibility for the organism, so therefore it is bipedal. And then C shape, the flexibility is not there, therefore it works on 4 limbs, quadrupedal. So the pelvic girdle for the case of bipedal must be short and wider to support the upper weight and allow easy movement. The other one, it must be small and narrow. Then foramen magnum for the bipedal organism, it must be in the forward position. That's how we want it, in the forward position. While the foramen magnum, it must be in the backward position for the case of the organism which walks on 4 limbs. Why is it supposed to be in forward position? Forward position so that the spine can connect to the skull vertically. And then now the skull is going to face straight in front. Straight in front. Hence, the organism is going to walk on 2 limbs. So if you look at this, it's a little bit in the forward position. Don't say in the center. Center of what? No. Even this side can be center if I count from I measure from this side to this side. So it's going to become the center. So the center, we say in forward position. While this one is a little bit backward. So it is not in the forward position like this. So it is backward. So it means that this is a bipedal organism. This one is the quadrupedal organism. So they're saying, give one visible reason for your answer. So the answer here is B. If the answer is B there, therefore it means that give one reason. It's because the foramen magnum is in the forward position. So it is in the forward position. And then they're saying that tabulate 2 visible differences between the upper jaw. Now here, they're looking only and specifically the upper jaw. These jaws which you see. So don't talk about other parts. The upper jaw in diagrams A and B. When they say the word tabulate, that's a mark. The word tabulate, even you can see that is 5 marks. So when you draw a table, please make sure that you draw a correct table so that you get all the marks. So you write here, for example, skull A and then skull B, for example. Make sure that you close the table. You lose marks here. Ne? Don't just draw a T and then you write. No. If you draw a table, make sure that you put the heading. Ne? Please close. Don't just draw it like that. Please close the table. Close the table. It doesn't cost you anything for you to close the table so that you don't lose a mark. Be neat when you are writing. All right, now let's compare. Skull A, skull A. This is skull A, skull B. So number 1, they're saying the upper jaw. Let's look at these teeth. This and this. The canine here, they are big, meaning that they are well-developed canines. Here, the canines are less developed, meaning that they are small. Number 2, this one has a rectangular palate. Here, it has a U palate. Ne? It's like a C or a U palate. Here, it is rectangular palate. So, those are some of the differences you can talk about because they are talking about the jaws. So the palate here is rectangular palate. Here it is U shape, or the jaw, they are U-shaped, and then here, yes. And then they are saying that so you get 2 marks for the difference and 1 mark for the table. Then they're saying explain the significance of the shape of the spine. I talked about it. That is associated shape of the spine, significance of the shape of the spine that is associated with the skull B. What is skull B? Skull B, we say that it is walking on 2 limbs, and the shape, if you look at it, it is S shape. Because they didn't talk about which shape is it, then mentioning the shape could make you to earn a mark. Therefore, the shape is S shape. Yes, S shape. But what is this S shape for? It allows flexibility of the organism. So it makes the movement a little bit easier. Then, in most cases, things related to the graph, I only point out what is very important and the most likely questions which are likely to come when they bring a graph, because so that there is no cramming about the graph. So now, if they ask you to describe, when they say you're supposed to read the whole question, Herbicides, herbicide is a chemical which kills a plant. Glyphosate is used to control the weeds in maize fields. The herbicide kills the weed, but it cannot kill the maize plant. The herbicide kills the weed, but it can also kill the maize plants. It has been found out that weed developed resistant to glyphosate treatment. Scientists carried out an investigation to determine the development of the glyphosate resistance in the weeds. The weeds were treated with the same concentration of the glyphosate each year from 2009 to 2016, and the percentage of the glyphosate resistant weed in the field was recorded every year. Okay. Results as follows. You see that glyphosate resistance, as year goes on, the resistance increases, but 2015 and 2016 the resistance became constant. So now I'm saying describe the change in the glyphosate resistance over a period of year. It means that here, whenever they ask you to describe, you tell us what is happening, because if you draw this on the graph, not a bar graph, just a normal graph, you will see that it will be continuing, and then it forms something like this. So there is an increase from 2009 2015. There is a steady increase, yes, in the resistance of the weed over the herbicide, and then it remained constant between 2015 to 2016. So it remained constant. So when you say description, you tell us what you see. Don't give us the reason why there is that increase. That is description. When they ask you to explain, then you tell us why, you tell us that there is an increase, and then you give us a reason why that increase is there. So that is what we call explanation. But if they ask you to describe in science or in here, you just tell us the shape of the what of the graph, or the trend, how it moves. That's what they want. That's why you see that the marks, even they're not that a lot. Then they're saying that calculate the percentage increase in the glyphosate resistant weeds from 2010 to 2015. Show you're working. Here, whenever they ask you to calculate the percentage increase, whenever they ask you to calculate for the percentage increase, this is what you are supposed to do. Percentage increase is equal to the final percentage increase is equal to the final figure, the final value minus the initial value over the initial value times 100. Yeah, that's it. So in this case, they say that from 2010 it is 20, and the 2010 is the initial value, and then to 2015 is 65. So I'm gonna say the percentage increase is gonna be equal to 65 minus 20, yes, divided by because the initial, it is 20, then times 100%. So the answer you get is gonna be the percentage increase. So that's how we calculate the percentage increase. So this is a rough thing how you can answer questions when you really sit in this paper, or any paper you're writing. You can really have no questions which are challenging. You are not going to see any new questions. All the questions you're gonna find in the paper are always the questions we ask you every day. Those are the questions you're gonna find in the paper. So there is no new questions, because we don't have any new science which has come in. So the curriculum remains unchanged for a period of years. So no new questions. Don't expect new questions in your exam. So please try and focus on, we have the same format of setting, so we cannot set outside the examination guideline. So scientists isolated the gene from for glyphosate resistance from the weed and used it to genetically modify the maize plant. Okay, explain the economic benefit of making the maize plant resistant to glyphosate. Because the glyphosate, now look, they are saying that herbicide is a herbicide which kills plant. Maize is also a plant, and then we use it to control weed in the maize plantation or the fields. So we have to make sure that this glyphosate cannot kill the maize. So the reason why they make this maize to be resistant to glyphosate, so that when you apply the glyphosate cannot kill the maize what? The maize plant. So that is the idea behind it. So I'm not saying that this is the exact question which is going to come, but when they give you such question and you don't have any idea, you can go back in the passage, most especially these questions which are involving of reading, go back, read again, and then identify. Then they're saying that draw a bar graph. Before I go to the bar graph, can you identify the independent variable here? You can identify the independent variable here. So is the weed dependent on the herbicide, or the herbicide depend on the weed? It means that the weed will depend on the herbicide over a period of years. That's why here you see that over a period of time or years. That's why you see here that glyphosate resistance, the resistance will accumulate based on the period of years, meaning that this herbicide will be resisted based on the period the weed has stayed in contact with the herbicide. Therefore, the resistance is depends on the contact time it has stayed with the herbicide. Hence, resistance becomes the dependent, and then the period of the contact becomes the independent. Okay, the bar graph. Now here, when you talk about the bar graph, you need to know the things we are looking for, so that when you go to the exam, you don't miss any question concerning about the bar graph, concerning about the graph. The first thing you need to know is the caption. Sometimes some teachers don't explain, they just mark, they put a T, they put a C, they put a S. They don't explain this to their students. But what does it mean? How do you answer this question so that you don't skip any mark? All right. So now they're saying that the bar graph should illustrate the results of investigation in the first 4 years. So, okay. Now, I'm not going to draw the actual graph, but I'm just going to talk about how you can draw this bar graph without stress. So years are always an independent variable, meaning that this is time. Time is always an independent variable. So independent is always on x-axis. Independent is always on the x-axis. And then the dependent one is always on the y-axis. I always tell my students that if you forget which one is on the x-axis, which one is on the y-axis, you just write here the word ID, your ID. Ne? Yes, ID, which means that you know the ID you are using. Independent, dependent. So this one is going to go on the x-axis. Then you here put ID, X, Y. So this one is going to go on x-axis. This one is going to go on the y-axis. So that's how you can remember the x-axis and the y-axis. So you just do this small box. I repeat. Ne? You say ID, X, Y. So meaning that ID, independent variable on x-axis, dependent variable on y-axis. The first column on x-axis, the 2 or 3 or 1 goes to the y-axis. All right. Now, if you are drawing a bar graph, make sure that you draw a straight line. Don't mind about mine. Straight line. This is how we mark. You write a caption. Never forget about the caption. So we give you a tick for the caption. Then after that, you write the X, you write the Y here, you write the X axis here. But when you are putting the value here, for example, in this case, they are saying yeah, and then the percentage, the glyphosate resistant weed percentage. So it means that the resistance is gonna be here. Yes, the resistance is gonna be here, and then units, it is in percentage. Then the time is gonna be here, which is year, and then the years, they don't put anything. So if you just say time in years or just year, then it's fine. It means that now they're gonna give you another tick for labeling the what? Labeling the scale. Not labeling the scale, labeling the axis. So if you label the axis X, they can give you a tick, or Y, they also can give you a tick, or they can combine the two. Now, you go to, after that, you go to the intervals. Please don't start with a 0 here, yet you want to start. If you come back here on the, you'll see that is 10, 20, 32. Okay, this one, it's fine. You can start with a 0. But if it is starting from maybe 100, and then you go to 120, you'll find out that some people, they start here 0, 100, 120, 140, 160. So the difference between 0 and 100 is not the same as from 100 to 110. So if you start from 0, make sure that, and you say a difference of 10, it must be 10, 20, 30, 40, 50, 60. It must be consistent. If you don't do that, you're gonna lose a mark. And then now you're gonna have to draw your bar graphs. It must be straight. Don't forget that these bars must be straight. It must be straight. It must be straight. Ne? So now the distance between these bars, I'm saying this is a rough distance between this space and this space must be equal. It might not be exactly the same because you don't use a ruler when you're marking. No, but when you look at with your eyes, you see that these distances are the same. So it must be equal. These bars, the size must also be equal. So that we give you, if the scale, the interval is the same, we give you a mark for the scale. Yes. If you have drawn the bar graph, not a histogram whereby the bars are this connected to each other, then we give you correct type of the graph. We give you a correct type of the graph. We give you the mark for the caption. Then we give you a mark for labeling the axis. We talked about it. Then we give you a mark, maybe 3 of them. We give you a mark for plotting if you have plotted this correctly. So that's how we mark the bar graph or any other graph of any kind of nature. So basically, that's how we mark. Then the last question is natural selection and also out of Africa hypothesis. Still here, I'm not going to answer the actual, like the way it is here. I'm just giving you the ideas how you can answer these questions without stress. Because when you are bringing the question, we can bring it in a different version. Yes, we can bring it in a different version so that you don't lose those marks. So when you modify, where do you modify and how do you modify it? A population of lizards on the island A were well situated to feed mainly on insects. So it means that these were feeding on insects. Scientists moved the 5 adult pair of the lizards species to the neighboring islands. They are saying the island A, island B, here they reproduced and a new population formed. They're saying the island B has large supply of plant with the fibrous leaves, and fewer insects. It means that now the food has become scarce. So this one is going to become a problem. Yes, exposure to this insect new environment may have caused the lizard to undergo evolution. Okay, they're saying 63 years later, scientists returned to the island B to conduct a further investigation on the lizard population there. They observed that the jaw size of the lizards had increased, and scientists also analyzed that the stomach content of the lizard found that it was mainly plant-based. They confirmed that the two populations still belong to the same species. They confirmed that the two populations still belong to the same species. Describe how scientists could confirm that they are describe how scientists could confirm that there was a change in the jaw size between the lizards of island A and the lizards of island B. So the question might not come like this, explaining this, but it can come in a different version. So we shall go and then see exactly where we need to have the answers from. So basically, what the scientists could use to confirm or could confirm that there was a change in the jaw is, this is an obvious answer. It means that they had to measure the lizards on the island A and the lizards on island B, and they check whether the jaw size is the same, and they can compare the differences if the difference is there. Then explain how the large jaw of the lizards on island B would be structurally suited to eat the tough fibrous. Explain how the large jaw of the lizard on island B would be structurally suited to eat the tough fibrous leaves. Yeah, this could be about the jaws being strong, meaning that the muscles are strong enough, and also the teeth, so that they can have a strong bite, and then they can chew that food. It means that the muscles, to do that, it must be strong, so that the force of grip is high, and then they are able to chew this kind of food. How did the scientists determine that the two populations of the lizards, both on island, still belong to the same species? Don't forget that a population, a species, these are organisms that can interbreed to produce a fertile offspring. So if they have the two populations and they try to allow them interbreed, and they produce a fertile offspring, therefore they can confirm that these are still the same species. But if they cannot interbreed to produce a fertile offspring, then automatically they are not the same species. But if they could interbreed and produce a fertile offspring, therefore it means that they are still of the same species. Explain the possible effect that evolution of the lizard has on the biodiversity. Explain the possible effect that evolution of the lizards has on the biodiversity. Here, it could be, it can increase the biodiversity or it can reduce the biodiversity. So basically, those are the two ways. If it forms a new species, then automatically it has increased the biodiversity. If it takes away an existing species, then it forms another species, then it means that it is decreasing the biodiversity. However much is creating, but it is decreasing because the other one is no longer existing. Or it could be 3 species, and then because now the food has come in a different angle, the 3 species, they modify into 1 species, or they modify, and the other ones, they become extinct. They become extinct, meaning that now they have also decreased the biodiversity. Use the Darwin theory of natural selection to explain the evolution of lizards with large jaws. Use the Darwin theory of natural selection to explain the evolution of lizards with large jaws. Here, the principle is the same. The principle of natural selection, whatever you explain, use the same principle. The first point you have to talk about is variation. There was variation among, now you have to tell us what they gave you, among the lizards, those with the large jaws and those with the smaller jaws. because the jaws are used to bite or to eat this tough food. Therefore, those lizards with big jaws, they could eat enough food, tough food. Therefore, they survived and reproduced in large number. Those with the small jaws, they could not eat enough food. Therefore, they died in large number. And this is called natural selection. Yeah, previously we are giving a tick on natural selection. This is called natural selection, but the chances are high that we no longer giving this marking point of this is called natural selection. But put it there, you never know that maybe we can review about it. This is called natural selection. So those with big jaws, they pass their genes to the next generation because they reproduce in large number. After many generations, all the lizards have big jaws. There is variation, I'm repeating, there is variation among the lizards. Those with, you tell us the variation. Those lizards with large jaws and those with the small jaws. Those with the large jaws, they could eat the tough food. Yes. And then they survived, they reproduced in large numbers, they passed their genes to the next generation. While those ones with the small jaws, they could not survive, they could not eat enough food, therefore they died in large numbers, and this is called natural selection. Those which survived, they passed their genes to the next generation, and after many generations, all the population of the lizards were made up of large jaws. That's how you can explain using Downey's theory of natural selection to show how this evolution of large jaws came about. Lastly, on this paper, please, you should never go to the paper without knowing these questions. The out of Africa hypothesis, which states that modern humans originated from Africa and migrated to the rest of the world. Don't forget the word modern. However much they might not give it as a ticket marking point, but in most cases, we criticize when the word modern is not there, because not all humans are modern. Homo habilis were humans. That's why you call it Homo, which means a man. Homo erectus were humans. That's why it's called Homo, is a man. Homo sapiens is a human. That's why you call it Homo, which means a man. So Homo habilis, the upright, the handyman. Homo erectus, the upright walking man. Homo sapien, the wise man. Those are, all of them are humans. That's why we are referring to modern humans. So please put the word modern humans. Homo sapiens, which means the wise man. I don't mean the school, I mean the man, you who is listening. Someone's going to ask me, what about us who are ladies? Yeah, is a man in science. All right. They're saying that name the family to which modern humans belong. They belong to a family called Hominidae. Hominidae. So they belong to a family called Hominidae. Yes, that's the family to which it belongs. They're saying that which genetic evidence is used to support the out of Africa hypothesis. We have two genetic evidences, but we only teach one. We have what called mitochondrial DNA and Y chromosome. But now we don't talk about the we don't talk about the Y chromosome. Only now the syllabus or the curriculum is talking about the mitochondrial DNA. So the evidence we give you here is mitochondrial DNA. Describe how fossil evidence is used to support out of Africa hypothesis. I've been telling these kids that these are the same questions we ask you. And if you check the book, our book we use, you can download even yourself. That's exactly how we mark exactly. This is the same way how they ask you this question. This book was released a long time ago before even these papers are being set. All right, let me answer. You have to talk about the oldest fossils which were discovered in Africa. For example, you have to talk about that Adipithecus remedus was discovered in Africa. You have to talk about that Australopithecus. Among Australopithecus species, we have Lucy, that is Afarensis. We have Taum child, that's Africanus. Mrs. Place, Africanus. Little fruit, Africanus. Carabo, Sediba. Ne? All those Australopithecus species were discovered in Africa. Homo habilis also in itself was discovered in Africa. That is a handyman. Now, when you reach here, you add the word the oldest. The oldest species, the oldest fossil of Homo erectus and Homo sapien, we also discovered in Africa. That's what you want. So you may not talk about all the Australopithecus species, all of them. You can just talk about one or two. So Adipithecus discovered in Africa. Australopithecus species discovered in Africa. For example, Taum child, Mrs. Place. Then you'd go to Homo habilis, discovered in Africa. Homo erectus, the oldest, Homo erectus and Homo sapien, discovered in Africa. Let me show you what I'm trying to say. This is what I'm trying to say. Evidence to support out of Africa hypothesis. There are three: the mitochondrial DNA, the cultural evidence, and the fossil evidence. This is what I'm trying to say. That fossils of Adipithecus were only found in Africa. Fossils of Australopithecus, e.g. Carabo, Little Foot, Child, Mrs. Place, all were found in Africa. Homo habilis were only found in Africa. Oldest fossil of Homo erectus and Homo sapiens were discovered in Africa. This shows that modern humans originated from Africa and migrated to the rest of the world. You see that even I highlighted the key points which you need to know when you're writing in your exam. Yes. So as I'm winding up, I will recommend you to use this book. Go to our website, download it free of charge. If you want to be printed, that's a different story. But go use these resources. I took time to write this book, and I wrote it with care so that every child of this country in South Africa must pass. You don't have a right to fail. You have a right to pass. You must demand for your rights. What is your right? The right of passing. Please, never ever demand for the right of failure, because you have the ability for you to pass, not the ability for you to fail. I wish you the best. Make sure that you go through these papers. You will make it. Thank you very much. Don't forget to subscribe, like, and share to others so that they also benefit in this before they write their final exam. Thank you very much. M.Saidi as usual. Welcome back to our session, which is guiding you for paper 2. We'll be looking at questions, most likely examinable questions, which are going to come in your paper 2, both in prelims and final exams. Don't forget that in most cases, we don't set new questions, but we use the questions which are prevailing, and then we modify them. So I'll be showing you how to answer these questions, and I'll show you different questions. Some of them I'll be just speaking about them, and then how do you answer those questions. So keep on so that you don't miss out anything, and don't forget to subscribe, like, and share to others so that you can also benefit. M.Saidi as usual. Let's start with the question 3. The diagram represents the portion of nucleic acids. Before I start this, don't forget that we have specific topics which we are supposed to look at in paper 2. The first topic is DNA, code of life. The second topic is meiosis. The third topic is genetics. The first topic is evolution, but both evolution in natural selection and then human evolution. So it means that these are the topics which are being set in paper 2. So when you are reading, you must focus on these 4 topics or 5 topics. So we're going to look at all questions and then the most likely examinable questions or questions you're most likely to find in your paper. If they bring a diagram like this, you have to find out which diagram is representing. So we represent, this diagram represents DNA. Why DNA? Because of this, the weak hydrogen bonds. And then it's two-stranded. What are some of the questions which are most likely to be asked about the structure of DNA? Apart from labeling the structure of DNA, they can ask you to describe the structure. Apart from labeling it, describe the structure of DNA, and they put there 6 marks, whereby you have to tell us DNA is made up of nucleotides. Each nucleotide is made up of a phosphate, a nitrogenous base, a phosphate, and a nitrogenous base, a sugar, and a phosphate. It forms 2 strands. Each strand forms 2 strands. Each strand is joined together. So you describe the characteristics of DNA. Basically, what DNA has, it forms 2 strands which twist around to form a double helix. So you can get the 6 marks from there. So in this case, let's label it and then find out what is a V. What is V? V is the nucleotide, because a nucleotide is a nitrogenous base, a sugar, and a phosphate. So this is a sugar, and then nitrogenous base, and then a phosphate. So this circle or box is showing you a nucleotide. Then Z is weak hydrogen bond. Use the word weak. Weak. We want to see the word weak. Then hydrogen bonds. Weak hydrogen bond. And then this is a nitrogenous base. Nitrogenous base. And then also this is a phosphate. Sorry, a nitrogenous a sugar molecule, which is, in this case, it is a ribose, deoxyribose sugar. And then Y is the phosphate group, as we saw it here. So now, since you know that this is a structure and you know how to label it, now let's talk about name the nucleic acid which is being represented. So we have seen it that this is DNA. And they're saying name 2 places in animal cell where you can find this DNA. Where can you find this DNA in animal? You can find in animal in 2 places. That is in the nucleus, and then in mitochondrion. If it's one nuclei, if it's one mitochondrion, if there are many mitochondria, if there are many nuclei, if it's one, it's called nucleus. So it should not confuse you. They are saying name portion V. They are looking for portion V. What is portion V? We say that these are the building blocks. This is the building block of DNA. So it is the nucleo. Molecule Y. Where is Y? Molecule Y is a phosphate. We have seen it that it is a phosphate group. Then they are saying that bond Z. What is bond Z? Bond Z is weak hydrogen bonds. So I think we have seen the answer here. Weak hydrogen bond, we have seen here, phosphate group, nitrogenous base, we have seen it, and then portion W, which is the nucleic acid. Sorry, which is the nucleotide. What is the natural shape? When they say natural shape, many students, they say helical, double helical. No, the only answer we accept in exam is double helix. Nothing else. You write something different from that, we won't give you a tick. Name the process during which this molecule makes exact copy of itself. How does DNA make exact copy of itself? How does it replicate? So the answer is DNA replication. So that is the process by which DNA makes exact copy of itself. Why does it occur? Some questions can come here. Why does DNA replication occurs? It occurs to increase or to double the chromosome number, to double the chromosome number. Where does it occur? When you talk about where does it occur, then you talk about the nucleus. And then when does it occur? It occurs during interphase. Yes. And then why does it occur? Why does it occur? We say that to double the chromosome number, to double the chromosome number. And then lastly, how does it occur? That is the process of DNA replication. If they bring a question like this, how do you answer? This is a DNA molecule. So if you look at it here, you see it is a DNA molecule. And why is it DNA molecule? Because of the thymine, which is indicated in the nitrogenous base. And then also the weak hydrogen bonds, 2 strands, so it identifies it to be a DNA molecule. And it cannot be RNA, because even here they say that this is what? This is a DNA replication. Yeah, so question them. Molecule W, where is molecule W? We have seen it, molecule W is here. It is a nucleotide, correct. Molecule U, where is molecule U? The whole molecule is DNA, correct. Then they are saying that Z is weak hydrogen bonds, correct. And then what is V? How do we know the nitrogenous base V? Because nitrogenous base is compatible to this or complementary to this, therefore, and this is given it here, which is thymine, therefore V is equal to A, which is adenine. And then they are saying that name the part of molecule W is W, X, and X and Y. X is the phosphate group, correct, and then Y is the oxyribose sugar. Why don't I say sugar? You have to be specific, because this is DNA, so it is the oxyribose sugar. Where in the cell does this process take place? Where do we find? We have answered this question that this process occurs in the nucleus. And then they're saying that name the phase of cell cycle where replication takes place. We have already answered it, and then we say that interphase. So at least when they bring such questions, you must be able to answer accordingly. So first try to answer what is being labeled. The moment you know what the picture depicts or what a picture represents, at least you are able to bring your mind and then you concentrate in that area. I'm not going to spend too much time here, only maybe if I see a very difficult question there, then that's when I can try to answer related to this, because I wanted to go to transcription and the translation. But molecule C is phosphate, and then this is a nitrogenous base, and then this is phosphate, sugar, deoxyribose sugar, and then nitrogenous base. It forms what you call a nucleotide. And then weak hydrogen bonds, and then this is a sugar this is a complementary base. So this is a complementary base. And then here, yes, they said this weak hydrogen bond. And then you have this. How do you call that? That is not a strand. We call it a backbone. And then the whole strand must include with the nitrogenous base and this. So the difference between a whole strand and a backbone is the backbone does not involve the nitrogenous bases. If you are asked, what is this bond? This is a phosphate sugar bond, but it is a covalent bond. If you refer it to physical science. Okay. Two scientists who discovered, James Watson and Francis Crick, name one organelle where DNA is found in the nucleus and the mitochondria. Describe how mutation of DNA may change the structure of the protein. The moment DNA molecule changes, is going to change the sequence of the bases on messenger, and the messenger RNA is going to change the sequence of the amino acids in which they are being brought to form a protein. So if a different protein is formed, if the amino acid leads to a different sequence, then a different protein is going to be changed and or is going to be formed. Then amino acid leads to the same sequence as previous one. Then the protein is not going to, a new protein is not going to be formed. Tablet 2 different structure difference. Now, if the major point here is structure, the word structure between the monomer, and they are saying about the monomer of DNA and RNA. So here they are talking about the monomer, and if you look at the monomer, they are looking at the nucleotide of DNA and the nucleotide of RNA. So you only look at the structure which is the nucleotide, of which the nucleotide is made up of a phosphate, a sugar, and then the nitrogenous base. So there are only two differences here you can look at. You can look at the sugar. DNA nucleotide has deoxyribose, deoxyribose sugar, while RNA has ribose sugar. And then DNA has thymine, while RNA has uracil. So after that, you draw a table. It must be a complete table. Yes, it must be a complete table. And then you put DNA, and then you put RNA at least. Then you get a tick here, a tick here, a tick here, a tick here, and a tick for the table, which makes it to be 5 marks. And if you find a question like this and tablet, and there is only 10 minutes or 10 seconds remaining, don't waste time. At least you can grab that 1 mark. Just draw a table like this, and then put DNA, and then put RNA. Yeah, even if you don't have anything here, then we shall come and give you a tick. At least you will score that 1 tick. So yeah, please try to use different ways in which you can get marks. Yes, yeah. So this is the same thing we have been trying to discuss. Phosphate, you have seen them, and then M2 process, which require the strand of the nucleotides in a single strand of DNA. Sometimes people think that DNA can be a single strand. It can be a single strand. What makes this one to be DNA is because of the thymine, which is being indicated there. So thymine is in this form, while adenine is in that form. Then they're saying that how many nucleotides are represented in the diagram? Yes, let's try to answer this. How many nucleotides? Every nucleotide is a phosphate, nitrogenous base, and a sugar. So this is 1 nucleotide, the second nucleotide, the third nucleotide, and then this is the 4th nucleotide. So the answer is going to be 4 nucleotides. And then they're saying phosphate molecule, then phosphate molecules, you just count them 1, 2, 3, 4. So the answer is also 4. And then they're saying that how many nucleotides, okay, I done that. Sugar molecule, there are 4, but sugar, this is the phosphate, and then this is the sugar. Write down the nitrogenous base from top to bottom as indicated by the arrow as complementary base strand and this molecule. Here is just T, they're saying on DNA. Complementary. So they are not talking about convert it into RNA. Don't forget that A goes with the T, then C goes with G. So whenever there is A, T, we put A. Whenever there is C, we put G. Whenever there is A, you put T. Whenever there is C, you put G. So basically, that's how it's supposed to be answered. Name two processes that require the DNA strands of DNA molecule to separate into single strand as shown in the diagram. Which processes? Two processes. We only find this in when the DNA replication is going to take place, and also we find this when there is a transcription. Remember here you form two strands, two separate strands. Each strand is acting as a template. While here you form two separate strands. Only one strand will act as a template. And while here two strands will act as a template. So these are the two processes which make DNA molecule to open. DNA profiling is also not a big deal. Technique, you have to know how to identify this. Read the question very well and then find out. Don't just answer the question. Detectives were investigating crime scene, found the blood on the broken window. Then they suspect that the blood was that of the criminal. To identify the criminal, they analyzed the DNA sample from different DNA sample from the blood and compare it to the four suspects. Now look, this is DNA sample, and then these are the suspects. It means that these are the DNA they found on the but you have to see where it is corresponding. This one goes with that, this one goes with that, still, yeah, this one is that. Use a ruler to identify this, that, this with this, this with this, this with this, this with this. So it means that Jenny is a suspect for that case, whose DNA profile is matching with the DNA sample obtained from the crime scene. Name the technique which was used. It's DNA profiling technique is DNA profiling, and what is being shown here is a DNA profile. So you must know the difference. DNA profile is not a technique. DNA profile is what you obtain when you analyze the DNA sample. So who was the possible criminal? We have seen it is Jenny, because the DNA profile, or the DNA profile of bars there of sample, DNA sample profile is matching with the profile of Jenny, or the band of DNA profile sample, they are matching with that of Jenny. So state one other use of DNA technique, or state other use of the technique identified in the question above, meaning that the DNA profiling, what is it used? Can be used to identify the relatives. They have talked about the criminalism, so you don't talk about that anymore. So talk about the identification of the perfect organ donor. You can talk about lost relatives. You can talk about diagnosis of the inherited diseases. So there are so many uses you can use to when you are dealing with the DNA profiling. So it's the same thing. You see that is the same thing. I don't usually take too much time here because these questions are really open. Who is a suspect? You know how we have identified it. What diagram is DNA profile? What does the diagrams above represent? They're not saying the technique, they're asking for the diagram. Therefore, it is DNA profile, not DNA profiling, because here they said which technique, name the technique. So it's DNA profiling, but here it is DNA profile. Which suspect? Look at the bands and then you match them, give a reason. So the suspect, if you look at them, you'll find out that Q is most likely to be the suspect, because the bands of broken glass, they are matching with that of Q. So they're saying that name 2 possible disadvantages of using this evidence in the court. It might not be accurate 100%, because number one, they can human error, number one, and also it may not be unique if a small sample is used. Also, a human being can manipulate them, so it may not be 100% accurate. So we have seen many questions concerning about DNA profiling. Download this and then try to do these questions. Where you get stuck, come back to me. Ask me questions. I'll be able to help you out. My number is always on the website 0848331738. Yes, and then speak to M.Saidi, who is always ready to help you out. Then another question is still the same tablet. Whenever you talk about tablet, make sure that you draw a nice table, and then so that you get that 1 mark. That's why there are 5, because they're asking for 2. They give you 2 marks there, and then 1 mark for tabulating. Describe the process that is being represented or responsible for formation of molecule B. So here, molecule B is messenger RNA. Why messenger RNA? It's because of this. So this is not, you see, this is messenger RNA, which is uracil. Therefore, and also 1 strand is being used. So that's evidence to show that this is transcription, because they are saying this is non-coding strand. So it means that only this is acting as a template. Yes. So they are saying that describe. So what are they looking for here? Describe the process that is responsible for formation of B. If you look at B, is messenger. So you describe. How do you describe here? Let me just talk about it. As I told you, talk about DNA double helix, unwind, weak hydrogen bond, break, and unzip. So when the weak hydrogen bond break, it's the same thing saying unzip. So you get 1 mark there. To form 2 separate, the word separate must be there, 2 separate strands. Only 1, you see it here, that only 1 is acting as a what? Is acting as a template. Only 1 is acting as a template. So it's being shown there that this one is acting as a template, and then using free floating messenger RNA. So you have to write messenger RNA nucleotide, yes, to form a complementary strand where adenine goes with uracil, and then guanine goes with cytosine. Adenine goes with uracil, and then guanine goes with cytosine. The complementary strand move out of the nucleus via the nucleopore to the cytoplasm on the ribosome. So basically, that's what you're supposed to explain. Please don't try to bridge it, because it's a process. 1 step leads to the other. If you start with what is on at the bottom and then you start with it, then you lose marks, because the sequence is not the same. So you cannot start with the last step and then you end with the first step. You have to start with the first step and then end with the yes. So molecule B, we said it is messenger RNA. Name the process is transcription. Explain how the protein molecule would be affected if codon GUU is changed to CGC. So if here it depends on 2 things. If there is a table, yes, now you compare the table and see if it's gonna lead to a different amino acid. But because now this is open, then we assume that it's gonna be a different amino acid. For example, if you say that you started with GUU, yes, and then GUC, from this to that. If this, let me explain this. If this is tryptophan, yes, codes for tryptophan, and this one also codes for tryptophan. However much this has been changed to this, is going to code for the same amino acid. Therefore, the functioning of the protein is not going to change. But if this tryptophan, and then now this one codes for example, leucine, then it means that there will be a change in the functioning of the protein, because this is tryptophan, and then this is leucine. Therefore, a different protein will be changed, will be formed, and then still the functioning of the protein will also change. So explain how the protein molecule would be affected if this is changed to this. So therefore, you tell us that if GUC is changed to GUC, GUU is changed to GUC, because they say that codon this changed to, because this, when they say codon, it means that we have started from the messenger RNA. Then this will lead to different sequence of amino acid. Different amino acid will be brought to the molecule or to the messenger RNA, not messenger RNA, will be brought to different amino acids will be brought to ribosome, and then a different protein will be formed. So that's basically you have to understand. And then when you come here, this is translation, because it is occurring on the ribosome. So what is A? A is messenger RNA. What is B is the ribosome. And then what is C? C is the transfer RNA. What is Z? Z is the amino acid. So this is the monomers, amino acids, monomers of the protein. And then this is the transfer RNA. You have seen it. And then this is the ribosome, and then this is the messenger RNA. So basically, you need to do that. And then what is this? These are 3 bases. So therefore, these are codons. Yeah, remember, the 3 bases on the DNA, we call them cods. And then 3 bases on the messenger, we call them codons. And then 3 bases on the transfer, we call them anticodons. Organelle X, what is organelle X? We have seen it. We have seen all the questions. You see now, when you start with trying to label them, you'll be able to answer all the questions. Molecule W formed, you see, organelle Z, X, we have seen it, ribosome, monomer W, the monomer. They're saying the monomer of molecule W. Remember the building blocks, that's what we call the monomer. Therefore, it's going to be the nucleotide. Be the nucleotide. Yes. And then molecule W and Y, W and Y, we have seen it. W is messenger RNA, and then Y is the transfer RNA, and then molecule or organelle X, we have seen it is a ribosome. So still the same thing. You can see some people, they call this a protein. This is not a protein. It is only a protein if it is 50 and above amino acids. So what is this then? This is a polypeptide chain. Yes, so it is a polypeptide chain. So what is X? This is the amino acid. And then what is this? This is transfer RNA. And then what is Z? We have seen them. These are anticodons, or anticodon. And then this is messenger RNA, and the 3 bases on the messenger RNA, we say we call them codons. You see, you can't fail such questions. When you come to the question, you see that they're asking you the exactly same thing, and then asking you describe the process of transcription, which we have described. So basically, at least you have some light on DNA, and then maybe we will talk about the process of translation, and then after that we wind up with this coding and then the mutation. These are some of the short question which you need to know before you go for the exam. Number one, DNA replication. You have to know DNA replication, the process by which DNA makes the exact copy of itself. Where does it occur? We have seen it in the nucleus. When does it occur? We say that it occurs during interphase. And then we said, why does it occur? We say that to double the chromosome number. And then how does it occur? That's the process of DNA replication. And don't forget that here you have to explain that here are the tools. You form two separate strands, and each strand is acting as a template, and that is the major difference between DNA replication and transcription. So in most cases, people confuse transcription and the DNA replication. Then number two, describe the location and the structure of DNA. Here I explained it at the beginning, the structure of DNA. In most cases, give it 6 marks. And then location, we have 3 locations, that is nuclear DNA, mitochondrial DNA, and chloroplastic DNA. So if they talk about in plants, then you talk about all of the 3. But if they talk about in animals, you only talk about the 2. That is the because in we don't have a chloroplast in animals. So describe the structure of RNA. Structure of RNA, it's a little bit the same as DNA, but only few things which are not there. That is the thymine, the double strand, and then also the weak hydrogen bonds. Describe the involvement of different types of messenger RNA process in protein synthesis. Here they are talking about the functions of each. For example, you talk about transcription and then translation. So it is like you are describing the whole process of DNA of protein synthesis. Another thing you need to know here is also describe transcription in detail, and then also translation also in, yes. And then also what they can ask you is why is the mutations which occur. And I say that if you change the code, for example, from GUA to GCA, then if these two they code for the same amino acid, therefore the protein will not change. The structure of the protein will not change. But if the different amino acids are being coded for, then the structure of the protein will be changed. So let's go to meiosis. Let's go to meiosis. There are so many questions which can talk about DNA. And also the base calculation, whereby we're supposed to talk about if adenine is equal to 10%, what is the percentage of guanine, or the ratio of guanine, whereby you have to tell us that adenine goes with the thymine, and then cytosine goes with the guanine, whereby if this is 10, or definitely this is 10, and then 10 plus 10 is equal to 20. Since this is percentage, therefore I'm going to say 100 minus 20, which means 80%. And then 80%, so what happens? So it's going to give us 80% for these two, so divide by 2, and then from 40%, therefore this is 40, then this is 40. So this is just a simple recall. I hope you remember. Please don't go without knowing it, because at least those 2 marks you'll be able to answer them. And then if they are looking for the ratio, you can put them maybe adenine now. Adenine, we said it is 10, and then maybe with the guanine we said is 40. So divide by 10, and then we're gonna get 1, 2, 4. So sometimes they swap it and they say 4 to 1, and then because when now you have seen 1, and then you have seen 4, you have seen 4, you have seen 1, you just circle that. It is what? It is wrong. So when you go to meiosis, so many point where crossing over takes place. We call it chiasma. You see, a chiasma. Splitting of the cytoplasm, we call it cytokinesis. Okay, we can answer them. Failure of the chromosome to separate during meiosis, the non-disjunction, the structure in the cell that forms spindle fiber, centrioles, and then the phase of meiosis where homologous chromosomes are aligned along the equator, that is mera phase 1, because they're saying homologous chromosomes. And the point at which the two chromatids of the chromosome join, we call it centromere, and this division of the nucleus. Division of the nucleus is karyokinesis. And then they're saying that the site of meiosis in a female, because in female we find meiosis in ovary. So you write ovary there, ovary, and then the exchange of genetic material between chromatids in the homologous chromosome, which is crossing over. And then they are saying the genetic disorder caused by extra chromosome on chromosome number 21, that is Down's syndrome. Yes. And then they are saying that the structure formed between the centrioles during the cell division with spindle fibers. Yeah, spindle fibers. And then they're saying that non-sex chromosomes in human, we call them non-sex autosomes. And then they're saying the condition in the cell where there is only one set of chromosome. How do you call that condition? We call that condition haploid. And then the structure that is responsible for formation of the spindle fiber during spindle fiber, we say that is called the centrioles. Yes, and then the A chromosome aligned along the equator, none, because here it must be mera phase. Random arrangement of chromosomes, it must be mera phase. So it's A, B only. The site of meiosis in plants, anthers, and ovary, both in, this is male, this is female, and then the process of meiosis produces result in 4 haploid daughter cells, 4 diploid. So this one becomes wrong, so this is the correct one. The mechanism to introduce genetic variation, crossing over, is correct. Random arrangement of chromosome is also correct. So it is both A and B. So basically, you see, yeah, so now we are starting the other part of meiosis. All right. Please don't forget to subscribe, stay in touch. If you have any problem, we need to smash these questions, and then we get all the marks at the end of this period. Yeah, when you come to meiosis, there are some questions we can talk about. Let's see what we can go through so that they can also benefit you in answering questions related to meiosis. I know it is a simple topic, but is a little bit challenging to some other. But let's see if we can smash these questions, and then we get a better understanding. If you don't understand the topic, let me know. I'll help you out so that at least we can have more distinctions in this year. All right. They are saying that here you are seeing diagrams. What comes into your mind is the kind of phases which this diagram is indicating. So what is C? C is the spindle fibers. What is B? This because there are 2 chromosomes of the same kind or the same size and structure, therefore is homologous chromosome. Then the joint between 2, or the joint between 2 chromatids of the same chromosome, it is a centromere. And then diagram A, what does diagram A represent? Because of this one is starting to form, so it can represent prophase 2. Why prophase 2? Why not prophase 1? It's because now this, they are individual, they are not in pairs. And then number 2, you see that crossing over took place a long time ago. And then this one is metaphase. The chromosomes are aligned. If you turn this, and then comes this side, and then this goes that side, so that this thing is in between your chest. So you see that if this is one side of the chest, and then this is on the other side of the chest, I think now you see what I'm trying to say. So you see that they're in the middle. So why? These are the spindle fibers. Yes, if this is the spindle fiber, if this is the spindle fiber, and this is the spindle, sorry, centrio. So these ones are in the middle. That's why you say that it is under metaphase. But metaphase, which metaphase? It is metaphase 1, because these chromosomes are in the middle. Then they say identify A. We have seen it. A is centromere. B, what is B? B, we have seen it homologous chromosomes. Then C, you see, we have seen it as spindle fibers. Identify the phase in diagram 3. Diagram 3, because they are separating. Yes. So if they are separating, then they are moving to opposite poles. You see them? They are moving to opposite poles. Therefore, it is anaphase. But which is it? Anaphase 1 or it is anaphase 2? Is it anaphase 1 or anaphase 2? Because now there are chromatids which are moving to opposite poles. Chromatids moving to opposite poles. Therefore, it's going to be anaphase 2. Anaphase 2. And then they're saying that if it was anaphase 1, then this would have been individual chromosome. This would have been a chromosome moving to the other side. Also, this one would have been a chromosome, and the centromere there, this would have been a holochromosome, and also this would have been a chromosome. So it would have been anaphase 1. But because their chromatids moving to opposite poles, therefore it is anaphase 2. Then they're saying that write down the number of the diagram shown in the sequence which the phase occurs. The number of the sequence. They're saying write down the numbers of the diagram to show the sequence in which. So basically, we have to look for meiosis 1. The chromosomes are supposed to be in homologous pair. So this one becomes this is homologous pair. So it is 1. Therefore, it is what? It is metaphase 1. And then this one is prophase 2. And then after prophase 2, they separate, which is, so it's going to be 1, it's going to be 2, and then it's going to be 3. So when you are writing the numbers here, it's going to be diagram 2, diagram 1, and then diagram 3. So it's going to be diagram 2, diagram 1, and then diagram 3. Then one difference between metaphase 1 and metaphase 2. In metaphase 1, the chromosomes are aligned along the equator in homologous pairs, while in metaphase 2, chromosomes are aligned along the equator singly. They are individual chromosomes are the one which align along the equator. So that is the major difference. And then if we see another question, so label it. Very simple. You see, we usually bring the same kind of questions. So this is the spindle fibers. This is the centromere. This is chromosome. Non-homologous chromosome is 1 chromosome. And then this is the centrio, which is being produced by centrosome. Okay, centrio. And then what causes these differences in the chromosomes or chromatids is because of crossing over. When does crossing over takes place? It takes place during prophase 1. How does it take place? Homologous chromosomes, they align together. Chromatids of the homologous chromosome, they cross over at a point called chiasma, and then they exchange the genetic material, and then they separate to form different chromosomes which are genetically different. Basically, that is the process of crossing over. You can't go to the exam without knowing the process of crossing over. Identify the phase of meiosis in diagram 1. Diagram 1, because now, yes, the chromatids are separating, is the same thing like this one. It's the same thing. The same thing like this. It's exactly the same. Yes. So it's gonna be anaphase 2. And then this one is going to be metaphase 2 because the chromosomes are aligned along the equator singly. Yes, they align along the equator singly. Then they're saying that identify the phase. You are done with that. State what happens to structure D in the next phase. Where is structure D? Structure D, these are the chromosomes. So the chromatids, they move to opposite pole due to contraction of the spindle fiber or due to the shortening of the spindle fiber. Name the process during which genetic material were exchanged. What was exchanged? We say that the process is called crossing over, and then we said it crossing over, and then we said it occurs during prophase 1. State the consequence if the mentioned does not occur. If this process does not occur, it means that organisms it will reduce what we call the genetic variation. It means that more organisms will be more similar to each other. So it will reduce variation among the organisms because crossing over leads to genetic variation. Then they're saying that give the number of the chromosome present in the original parent cell in this diagram. They didn't say this is a human cell. I don't see anywhere there saying this is a human cell. So you have to know, you have to use the number which is being given. So because this is meiosis 2, and meiosis 2 leads to the diploid state being halved. So for example, you start with 1N, and then you form 2 cells, which is 1N, and then another 2 cells you divide to form 1N, 1N. So if you start with the 4 here, therefore it's gonna be 2, it's gonna be 2, it's gonna be 2, it's gonna be 2, it's gonna be 2. So it means that this is meiosis. Therefore, this is 1, this is 2. Therefore, the total number is 2. Please don't talk about 46 if they didn't say that this is the human cell. And then now they are saying original cell. So if this original cell, therefore it's gonna be 4. And then you see now they are saying, specifying it, human cell in the sample diagram 2. Because diagram 2 is haploid, therefore it's gonna be 20. It's gonna be 23 chromosomes. So basically, that's how you're supposed to answer such questions. So you can answer these questions, at least in meiosis. What is A? A, in most cases, students confuse it with a nuclear membrane. This is not a nuclear membrane. You can't have a nuclear membrane when the cell is under a metaphase. Therefore, this is the cell membrane. This is the chromatid. This is the chromatid. This is the spindle fiber. And then this is the centriole. So this is metaphase 1. While this one is anaphase 1. Why? Because chromosomes are separating. What about this? This is the telophase 1. Why? Because they are chromosomes. They are chromosomes which are still double-stranded. Yes. Or at the beginning of prophase 2. Yes. So you see that now once you name that, you are able to answer this question. What is phase 1? We have seen it is metaphase 1. What is 3? We have seen it is telophase 1. Then you're saying the letter only of the part contains DNA. Give the letter of only part that contains DNA. It is B, because in this case they didn't mention any chromosome here. It's only B, which is chromatid, and then attaches the centromere of the chromosome, attaches the centromere chromosome, which is C, attaches is the centromere. Yes, and then they're saying that forms spindle fiber, which is the centrioles. Where is the center mentioned is D. You see, we are able to answer these questions. Name the organs in humans, male, where meiosis occur. In humans, male, it occurs in the testes. While in female, it occurs in ovary. In plants, it occurs in anthers, that is male, and then stigma, ovary in female. So here they produce sperms, they produce ovum or ova. Here they produce pollen grain, while here they produce ovules. Yes. So basically, that's a simple illustration of meiosis occurring in plants and also in animals. Describe how meiosis leads to genetic variation. So now here you are talking about only processes which talk about genetic variation. Here you talk about crossing over. So you describe crossing over. Then you talk about the random arrangement of chromosomes during metaphase. Metaphase. Yes, so basically that's what you need to explain. It can lead to genetic variation. You can also mutation can also be part of meiosis. Why? Because if you talk about Down syndrome, it occurs during the process of what? Occurs in process of meiosis. But these two, they can help you to get those marks you are looking for. Here is an example of a karyotype. You have two individuals. Identify which karyotype. Here, when you're given a karyotype, look at, is there any abnormality? Which one is the male and which one is the female? So if you look at this, no abnormality. All of them, they are in pairs. But if you look at that, one is long and then one is short. Therefore, this is X. This is X and then this is Y. So therefore this is a male. And then this, both of them are the same size. Therefore this is a female. So now from there you can now go and then try to answer. What term is given the chromosome number 1 to 22? So we call them, because they can ask you to describe the structure of a karyotype. You call them orosome. The orosomes. And then 23, these are gonosomes. Why? These are sex chromosomes. Yes. State the gender of P. We have seen it. P. Where is P? Male, because of this. Give one observable reason, because the chromosome number, the chromosome number, the chromosome pair number 23. One chromosome is bigger than the other. Therefore, one is X, one is Y. Hence, the person is a male. Then they're saying give one observable. We have seen it. Each of the pairs shown is a homologous pair of chromosome. State the origin. State the origin of each chromosome in the homologous pair of the chromosome. So here, where do they are two. Where are they coming from? These pairs, one is coming from the father, and then one is coming from the mother. Yeah, that's why there are two. That's why we say that when the sperm comes, is haploid, and then fuses the ovum, which is haploid. So one chromosome, another chromosome, when they combine, you form the pair. So it's the same thing. Sperm, ovum, sperm, ovum, sperm, ovum, sperm, ovum, like that. So one comes from the father, one comes from the mother, and then from a diploid, a zygote. Then they are saying that the diagram below represents the distribution of chromosome 21 as it appears in the gamete. So it's the same thing, guys. Yeah. So you see that you'll always find almost the same diagrams, so same structures. So now draw a diagram to show the cell during anaphase 2 of meiosis. Now here, when I ask you to draw a diagram, you must write a title. That is the first thing, title. You cannot draw anything without a title. Use a sharp pencil. It must be neat. Yes. Someone is going to ask you, am I neat when I'm writing? Yeah, I'm saying you must be neat. I'm not a student. All right. I'm saying that. So when you draw, they're saying this is the prophase 2. Prophase 2, why? Because they are single. So what do? You draw the cell, and then they're saying during anaphase. So it means that before it's going to be like this, and then the short one down. So this is prophase. Just drawing a structure, but you have to show the variations when I'm drawing. So during anaphase, it means that one, and then one has a variation. So it's still moving to the poles, and then also then the short one is gonna be the short one, and then it's also moving, you see, but it also has the biggest variation, and then this other one is completely shaded. You see, you have to show that. And then this is spindle, this is chromatid moving, and then and then this is the cell membrane. Then write the title. At least you are able to answer such question. Yes, so that when you come, we'll maybe give you a mark for title. Yes, a mark for drawing, correct drawing, and then maybe those are 2 marks. Now maybe 3 marks for labeling. Yes, so that's why I think labeled diagram. So meiosis is also easy. If you look at most of the questions, you will see that we have answered these questions. Can you answer this? Yes. 10 seconds, 8 seconds, 7 seconds, 5 seconds, 3 seconds, 1 second. Okay, let's see. Then they're saying that this is what? Spindle fibers. What is this? Homologous chromosome. What is this? Centromere. Yes. So these are homologous chromosomes. And then this is the centriole. And then this is the cell membrane. So you can answer these questions. You see, nothing which is going to be new in your paper. Name the process diagram above. You see, it is metaphase. You see, you have to turn it so that these things are in the sides. Check. You see, they're in the sides. So that you see now clearly it is metaphase. Name the process, name the phase, represent a diagram. We have seen it. Then they are saying name part C. We have seen it also. Centromere. Then B represent homologous chromosomes. B represent what is homologous chromosome? Chromosomes which have the same structure and the same size, and then they carry genes. Genes of the same characteristics are always on the same locus. So we call them homologous chromosome. Then they are not genetically identical, but genes of the same characteristic, they are in the same locus. Locus is a position of a gene. It's singular. Loci when they are many. Explain the appearance of chromosomes in diagram. Why are they shaded like this? Is due to crossing over, which took place in prophase 1, whereby the chromatid exchange the genetic material. That's why they are like that. They are saying that total number of DNA is the total amount of DNA is 12 viral units each daughter cell at the end of this cell division. How many DNA was in the parent cell? So they are saying that the total number of DNA is 12 units in each daughter cell. Remember, you start with the 2n. You start with what? 2n. Yes. So 2n, you form 2 cells, 1n, 1n. then you form another 2 cells, you form another 2 cells, so 1N, 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 1N. So if you started with the, if your daughter cells are having 12 here, so it means that then if you want to get the parent, is times 2, so which is 24. So the answer is going to be 24 DNA binary units. Don't forget to put the units as they are being given to you. So many questions, you'll find them like this. This is the stereokaryotype. Male or female is a female, while chromosome number 23 is having chromosomes which are of the same size and the shape. See? So describe the karyotype of person with Down syndrome. So a karyotype with Down syndrome will have an extra chromosome on chromosome number 23. Yes, so the pair chromosome, so there will be 3 instead of what? Instead of 2. So you see that these diagrams, these questions, you can answer them very well. Yes, you can answer them very, very well. All right, let's just do few questions about it. What is this? What is this? This is a centromere. What is B? Is a crossing over point where crossing over takes place. Therefore, is chiasma. These 2 chromosomes under crossing over, we call it a bivalent. Bivalent or homologous chromosomes during crossing over. Then this, what is this? This is a chromatid, and then what is this? Is a chromosome. So these are sister chromosomes. So now you can answer the question. Describe the process that occurred at D. So it means that they're asking to describe the process of crossing over. I explained it before. This is where tricky things come from. They're saying the certain number species has 42 chromosomes. They're not saying it's human. So don't talk about 46. Yes, please don't talk about 46. They are saying 42. The muscle cell, its muscle cell, remember muscle cells, those are autosomes. They're always 2N. Yes. Give the chromosome number of each of the sperm cell. So it's going to be half because sperm cell is 1N. Therefore, the answer here is going to be 21. The skin is autosomes is 2N. Therefore, it's going to be 42. And then ovum of the female of the counterpart is the same thing, is going to be 1N, which is 21. In most cases, the number of chromosomes are the one which are confusing students and they end up losing marks. So it's the same thing here. Crossing over took place. They can ask you to draw the diagram after crossing over. So members, students, please don't lose marks. Don't lose hope. You will make it. You will make it. I'm going to come back for genetics and inheritance. And then after that, I will go to human evolution. Thank you very much for watching. Don't forget to subscribe, like, and share. Don't also stop to comment so that I know where exactly I should put more effort and where to help so that you can get your distinction. Man with blood group AB, and then a lady who is heterozygous for blood group B plan to have children. Yes, they plan to have children. How many alleles control blood groups? You see this booklet for the distinction material. I've already addressed these questions several times. Different blood groups result in multiple alleles. Blood group, there are 3 alleles, alternative form of a gene. Yes, 3 alleles, meaning that they're occupying the same locus. However, only 2 alleles can be presented into a single organism. See, so the question, how many alleles? There are 3. Allele for A, allele for B, allele for O. So these are the 3 alleles. So there are 3 alleles. There are 3 alleles. I always tell students that before you go to the paper, you must answer these questions because at least one question will come. How many for blood group, if the blood group comes? How many alleles? How many alleles? Yes, we said there are 3. That is allele for A,
allele for B, and then allele for O. Please don't modify these alleles. When you say allele O and you put like this, we're gonna cross it. Please don't modify it, and don't say allele for A. We will cross it. This is a Roman figure. This is not I. It's a Roman figure. Allele for B, you write like this. We cross. So make sure that you write capital I and then you say A. You write capital I, you say B. You write small I or small I like that. Please don't modify them. Yes, because if you modify them, you will get 0. You will get what? You will get 0. Then we are saying that they're saying how many alleles. We are done with those alleles. Saying that describe how many. Okay, describe the blood dominance that occurs in inheritance of blood group B. Blood dominance, we have three types of dominance. We have complete dominance, codominance, and then incomplete. Incomplete, it means that, let me just show you with a booklet so that we don't waste too much time. Incomplete, look, this is, the moment you see a small error, it means that that is the complete dominance. One allele is completely dominant over the other. Then we have what you call codominance, which is A and B. Both alleles are shown. And then you have the recessive. Yes. So when you talk about the three types of dominancy, this booklet addressed it very well. There are three: complete dominancy, yes, incomplete dominancy, and codominance. Complete dominancy, one allele is completely dominant over the other, meaning that you have a capital error and a small error somewhere. Incomplete, both errors are dominant, are capital errors, but the difference, even in codominance, both errors are dominant, but the only difference which is there is that in incomplete dominance, one allele, sorry, here you produce an intermediate. For example, if you for example, here this is complete, you see, at least you have one allele which is recessive. Incomplete, when you combine the two, yes, like here, when you combine the two, you produce an intermediate. You see, an intermediate color, white and red, you produce an intermediate color which is pink. While in codominance, instead the cross is the same, but here you produce a mixture of the two individuals originally. So now when we come back here to the blood group, they're saying blood group B. What do we say? So it's gonna be blood group A, blood group B. Yes, remember blood group E can be homozygous dominant, or it can be heterozygous. So because of this small error here, this small error, it shows that that is a complete dominancy for blood group B, but blood group AB is codominance. You see? Yes. So here it is incomplete, so is complete dominance. This allele for B is completely dominant over allele for I. Hence, blood group B, the type of inheritance which occurs there, the type of dominance which is occurring there is complete dominancy. Complete dominancy. Yes. Use the genetic cross to show the possible genotypes and phenotypes of their children. Hey, from where? A man who is blood group A. So you see, you're going for the same procedure, and I've showed you how to mark this. You come here and say P1. P1 means parent 1. Phenotype, and then you tell us. I told you that you always get this from the question. Is the same man with blood group AB. So I come here and say blood group AB. Blood group, yeah, you write it. Blood group AB, and then crossing with the blood group B. Blood group B. Yes, blood group B. Yes. Then genotype, genotype. AB is obvious. There is no issue with that. So you're saying that blood group AB is A, then B, crossing with. Here is the issue: is it homozygous or heterozygous? They have to give you a clue which shows that it is homozygous or heterozygous. We go back to the question. Lady who is heterozygous, you see, they said it, they gave a clue that is heterozygous. So I'm gonna come here and I say B, small letter, and then I'm gonna say meiosis. Meiosis is gonna occur, and then I'm gonna have A, and then I'm gonna have B. Please leave space here, and then here also it's gonna be B and then small letter. And then I always tell you that circle them so that you don't get mad at anywhere. So you're saying that this is gametes. So after that, you tell us you go to uh, you go to crossing. Yes, you can use a Punnett square or you can cross direct. A, and then this is B, and then this with this is gonna be A, I, and then this with this is gonna be B, B, and then this with this is gonna be B, I. So how do you call this? We call this is fertilization. And then how do you call this? We call this offsprings. And then how do you call this? This is F1 generation. So we will come and then start marking. I told you P1, F1, we give you a tick. That's Mahala question. Mahala mark. This is also another Mahala mark. This is also another Mahala mark. So out of 6, at least you have 3 marks Mahala. Mahala, mahala, mahala. So you see, you can't go with once they bring a genetic cross, any genetic cross, you have 3 marks, mahala. Then they give you a tick there, they give you a tick there, they give you a tick there. What is the question? Yeah, genetic cross show the phenotype and genotype of their children. So the phenotype, this is the blood group. You have phenotype, blood group AB, you have AB. Yes, you have A, you have B, and you have, this is AB, this is A, and then you have 2B, 2B, 1AB, 1A, and then 2B. So, and then the genotype are these ones. You can write them again. So that's how we answer questions concerning about blood group. How many? Let me know. These are the questions. Let me know in the comment below. Now, people who are on live, how many alleles control blood group? Question 1. How many alleles control blood group? Let me know. How many genes control blood group? How many? I want to see the answers. I'm waiting. I want to see the answers. How many alleles control blood group? How many genes control blood group? Those are 2 questions I wanted to hear from you. How many alleles control blood group? How many genes control blood group? Let's go to the last question as I'm waiting for your answers from your side. Black leopard frog can be spotted. Okay, can be okay, sharp. Spotted frog, which is a runner, and then allowed to interbreed, and then they produce 150 spotted offspring, and 50, meaning that the ratio is 3 to 1. Obviously, if it's 3 to 1, remember when you're talking about this, we are talking about dominant alleles. These are dominant and these are recessive. So it means that these ones are the one which are dominant and these ones are one which are recessive. So which genotype? They have said it. Spotted frog were allowed to interbreed and they produced 150 spotted and 50 offspring without spots, meaning that this is the one which is spotted. Yeah. So frog, yes. they're saying that which genotype is, so this is the one which is dominant. Explain why, explain why, explain your answer. Yes, the answer can be explained based on the ratio you obtained, that you got 3:1 ratio, meaning that the dominant ones are more. Remember, when you are crossing them, you have three options. It can either be this, or it can be this, then you have that. So in most cases, these are like that. Just give me an example. Becomes 3, and then this becomes 1. So if it's numbers, they say this is 150, this is 50, and then 150 and this is 50, divide by 50, divide by 50, you get 3:1. That's where I got this ratio from. And then they are saying that a frog that is heterozygous for spotted black was crossed with a frog without spots. Spotted black, spotted. I said the spotted one is the dominant, crossed with the without spots. But they are saying heterozygous. Use D. Expect genotype and phenotype of F1 generation. So you see, the answer is simple, the same way we have been doing. So basically, you do P1 F1, P1 F1, you write the phenotype, you do it for me, get the phenotype from here, then write the genotype, you do it for me. Where do I get the genotype? They said spotted, they said heterozygous. It shows that spotted is, so meaning that if they are saying spotted is going to be heterozygous like that. So if this is like that, and then without spots we said is recessive, so basically is always homozygous. Then meiosis is going to occur, then split them, then you get the gametes, then fertilization. After that, then you get the offsprings here. Yes, so you're gonna do it for me. When I asked the people to tell me about, and then I also tipped them that this is not a frog, and hey, people were chasing you, are running away from. Okay, yeah, I was just joking. This is a frog, né? A frog, and how do we differentiate between a frog and a toad? Yes, a frog is a runner, and then a toad is called a buffo. So a frog is that one which can jump. You even see it's small in size. It can jump long distance. And then a toad, just the one which hops. Toads that doesn't jump. The one you usually see is they're just toads. Ne? Yeah. And then the other question, as I'm winding up, the other question is how many genes control blood group? Is one gene. And which gene is that? Blood group. And then how many alleles? Alleles are 3. Allele for A, allele for B, and allele for I, for O. And then don't forget to write them correctly. Allele for A, allele for B, and allele for yes. And then how many genes? Is one gene for blood group, yes. And then how many genotypes? The genotypes are 4. That is blood group A, blood group B, blood group AB, and blood group O. And how many genotypes? These are phenotypes. And how many genotypes? Genotypes are 6. That is blood group A. Remember, blood group A can be homozygous or heterozygous. Blood group B can be homozygous or can be heterozygous. Blood group AB is codominant, blood group AB, and then blood group O, which is recessive, making them 1, 2, 3, 4, 5, 6. Yeah, I think that is it. The next class is going to be genetic engineering. Don't forget to subscribe, like, and share for others for our daily new videos. There are some people who want to join my private classes, as now it is open. Private classes. please find the link in the description below. Yes, uh, join. You pick join class. Yes, so that you join the class. You get you ask questions anytime. We will try to answer you, but late night we might not answer you, but we will answer you immediately when it is morning. Yes, we'll help you with this life science and then physical science. People were asking me about agriculture. Agriculture is also coming, and also people were asking me about uh, maths. We have not yet. We do live classes, but we have not yet started the private classes. But until the teacher say that okay, it's fine, they can create space for you. So, but the live classes are always there. Here, when I talk about the private classes, it means that maybe you are in class and then you have a challenge. We have sometimes Zoom and Microsoft classes. Sometimes we attend from our website. Yes, please don't forget to share, to like, to for others so they can also benefit. It is M.Saidi as usual. It has been a wonderful uh, discussion with you. Thank you very much. See you again. God bless you. Don't miss out. Okay, they're saying that state the genotype of Z. Here, the way how we teach dihybrid cross is not the same way how we ask questions concerning about dihybrid. We don't ask you to explain everything in the dihybrid. No, we only ask you few questions on the dihybrid. So, please take note of that we don't ask you everything on the dihybrid. Okay, now the question is saying that they are saying that state Z is this with this. So basically, I'm gonna just draw a line. This you write what's there, and also is this line what's there. So it means that I'm gonna say small f, and then capital H, small h. Yeah. Why didn't I start with the capital H? Because these are two different alleles. Yeah, these alleles, one allele does not interfere the existence of another allele because these are different genes. Yes, Mendel's law of segregation. Mendel's law of, yes, independent assortment, not segregation. Mendel's law of independent assortment. The one allele of one gene does not influence the other allele, meaning that if you have big heads, a big head, that is not connected for you. Why do you have small eyes? So the allele, a gene for the big head, does not influence small eyes or the type of eyes you are having. So they are totally different. That's why I didn't start. But if it is the same allele, H and H, then automatically I cannot say H, capital H. No, I have to start with the capital letter. Yes. Okay, now let's go to our question. The question is saying that give the genotype of parents. How do I identify the genotype? This is a very challenging question to kids. But let me just show you how you can get the genotype of the parent because they didn't give you anything here. But look at the gametes. Pick a letter which is not repeated, then write it down. The first one is capital F. Capital F. Make sure that you don't repeat the letter before because there's supposed to be two alleles for each character. But first write the letters which are not. Don't repeat the letter before you complete. It's capital H. Capital H. We have talked about capital F. Capital F. Small f. So I add the small f. But here is small f again. So I've already written it. Then I go to H, capital H, capital H. I've talked about the capital H, small h. So I've got the genotype because they are supposed to be 4. And then when I come here, is still capital H, capital F. Yes, then small f. And then capital H, and then yes, small h. So yeah. So it means that both of them, they have the same kind of what? They have uh, the same kind of allele. They are heterozygous for their conditions. So all of them, they have the same kind of what? Condition. Then they are saying that number of genotypes that could result in order in offsprings with short fingers and continuous hairline. Yes, number. Uh, number. So in this case, we are going to say number of genotype that would result in offsprings with short. Let me underline this. They're saying short, but short, what did they say? Is short is the capital F, is dominant. So I'm gonna look for anywhere I have capital F, yes, and then they're saying that fingers, continuous hairline. Continuous hairline, we say that is recessive, meaning that if it's recessive, small h, but it cannot be recessive, cannot show in the genotype unless these two are being shown. They are showing. So how does it show? It's supposed to show by being recessive. So because if I have capital H and then small h, then automatically this individual is not going to be continuous. It's gonna be this widow's what? Peak. So automatically it's supposed to be small letters, small letters. So I'm going to look for where there is a small letter, and also I'm going to look for where is what? I'm going to look for where is a capital H. So once I get that, then automatically I'll be able to identify how many individuals are there. So I'm gonna say capital F, small h. This is the first one, the first individual. This is the second individual. Is there anyone else? Yeah, the third individual. Is there anyone else? No. So there are 3. So the answer is going to be 3 individuals. Then they're saying that allele for continuous allele is, remember, is caused by recessive allele. So they are not saying alleles, saying allele. They are not saying genotype, because if you say this is the genotype. They are saying allele. Remember, it's caused by recessive allele. You see, this is caused by dominant allele, meaning that these are recessive alleles. So please do not write 2. It's supposed to be 1, because even they're here, they're saying allele. Phenotype of a child who is homozygous recessive for, they're saying phenotype of a child who is homozygous recessive for both characteristics. Is here long finger and continuous hairline. So that is it. So this is a dihybrid cross. So if they ask you which type of cross is this, is a dihybrid cross. Next question. The next question is also about a dihybrid cross. Dihybrid cross. Dihybrid cross. What does it mean? What does it mean? Dihybrid cross. I hope you are seeing clearly. There is variation in characteristics, yes, of fur color and texture in the cats. All right, I'm seeing, and then they are saying that the table below shows the allele that control these two characteristics. Ne? These two characteristics. All right. What is next? They're saying that you have these characteristics. The Punnett square below shows the inheritance of alleles in the genetic cross. All right, here is the Punnett square. Name the dominant allele for fur color. Here you come where you see capital letter, it means the dominant allele. So this is B. So the answer here is going to be black. This is black. And then the recessive phenotype for fur texture. Recessive is going to be smooth. Smooth. So you see how the hybrid is being set. It's not a complicated thing. It's just a normal thing. It's just for you to see that you can answer this. You can answer this. Okay. So they are saying that uh, uh, we are saying that give the genotype of offspring X. X genotype. They are saying genotype. So if you are saying genotype, then it means that you're going to come here and here. So it's gonna be small b, capital R, capital R. So that is the genotype for that individual. Yes. Then they are saying that the phenotype of female parent is female parent, this male and this female. They're saying the phenotype is capital B, capital R. Capital B is black and the small r, small b is white and capital R is rough. So it's gonna be white and rough. White genotype of the parent. The genotype of the is gonna be white and rough. But I have skipped this. The genotype of X, yes, I have talked about it. So that is the answer for that. The genotype of parent male. Now you come here, male. They want the genotype. So how do I identify the genotype? How do I identify the genotype of this parent male? So I can only do the genotype of this parent male when sorry. I can only, as I taught you that what you do is you come here, say capital B, yes, you don't repeat it, yes, you don't repeat it, capital B, small b, yes, and then you go capital R, capital B, yes, you don't repeat capital B, but you have a small b here, put a small b, then you capital R, you look for, you have a small r, so small r, so that is going to be the genotype of that individual. Yes. Then they are saying that what is the genotype of the parent male? So you are done with that. State all the possible genotypes. The possible genotypes of what? State all the possible genotypes of offsprings of this genetic cross. Of this genetic cross. All possible state the phenotype of all offsprings of this genetic cross have in common. They're not just saying offsprings, they're saying what they have in common. Okay, come here and see. This is okay, capital B. Have a capital B. This one has no capital B. You see? Yeah. So no. So it can't be capital B. Capital R. Capital R. This one has a capital R, né? And then also this one has a capital R. So basically, state the physical, the phenotype of that all the offsprings of this genetic cross have in common. So definitely here I'm gonna say capital R, because we say that all of them they have capital R. It has capital R, it has capital R, capital R. I come back here and I ask myself, what is capital R? Capital R is a rough. So basically, what they have in common is rough. Yes, that is what you can do. That is the dihybrid cross. So we have seen questions of dihybrid cross. So they are saying that brown enamel of the teeth is sex-linked trait. A dominant allele on X chromosome causes brown teeth in human. So it means that if you have brown teeth, yeah, you know what is happening. It means that even if you go teeth whitening, it is brown teeth. Anyway, that's genetics. Let me know which kind of teeth you have. Anyway, brown teeth, we are saying that is caused by a dominant allele, but they have not given us any allele we are supposed to use so far. Explain why more males than females have white teeth. Remember, brown teeth is caused by a dominant allele. You see, it means that if I'm going to use B for brown, yes, and then small b for white, because it is caused by a dominant allele, and then it is caused by sex. It's a sex-linked trait. It means that females, the female is gonna be like this, yes, or it's gonna be like this. So that is a female who is brown teeth, while a male is gonna be capital B only. Then still the female can be this white teeth, while male is gonna be that. It means that males have two options, while female have three options. So, and even if a recessive allele appears on X chromosome of the female, it can be masked by a dominant allele. So it means that this individual is still able to live without that, without the brown teeth. So what can you say? How are you going to answer this? Basically, you know that we say that males have one X chromosome. Yes. Look at this general description. Males have one X chromosome. Yes, you see, one X chromosome. If a recessive allele appears on that X chromosome, the individual is having the condition, while the females have two X, you see. If a recessive allele appears on X, on one X, it can be masked by a dominant allele. The individual is still brown, not what? White. So for the lady to be white teeth or that sex-linked disorder must have two recessive allele, but males only have one recessive allele. So it is very common in males. So based on this question, how can you answer this question? You can say that males only have one X chromosome. That is a tick. Males have only one X chromosome. It's a tick. Or males have Y chromosome, meaning that they don't have X chromosome. Yes. And then what do? They have to inherit one recessive allele for them to be white. You see, they only have to be with one recessive allele for them to be white. But remember, the females have to be with the two. If they have one, they can be masked by dominant allele, and then the individual is what? The individual is brown. So males have one X chromosome. If a recessive allele is inherited, yes, the individual will have white teeth. You see? While females have two X chromosomes. Look, they have two X chromosomes. Yes? They have to be with the two recessive alleles for them to be white. Can I repeat? Males have one X chromosome. If a recessive allele is inherited, yes, the individual is white teeth. While females have two X chromosome, if a recessive allele is inherited, it is masked by a dominant allele on the other, meaning that they have to inherit two recessive alleles for them to be white. Yeah, please take note of that. We can bring it in different versions. Please don't go off. We can bring it for hemophilia. We can bring it for color blindness. So we can bring it in different versions. Yes. So that is the explanation for those marks, form marks. Then they are saying that a man with brown teeth marries a lady with white teeth. Which kind of teeth do you want? You want brown teeth or you want white teeth? Eh? You want white teeth or brown teeth? Okay. Use the genetic cross to show the possible genotypic ratio of their children. All right. What do you do here? Please do not forget that this is a genetic cross. You have to follow all the procedures. You have to all the steps because we mark by steps. You start with P1. P1 means parent 1. Parent 1, you have to tell us the phenotype of the parent, of which this phenotype is always given in the question. A man who is brown teeth. So you can say brown teeth guy is crossed with marrying a woman who is white teeth. Then they are saying that white teeth lady. Ne? Yes. They're saying that what happens? Use a genetic cross, use XB. So we go to phenotype, sorry, the genotype, the description of an organism in terms of genes. That's the genotype. The description of an organism in terms of what you see, the physical appearance of an individual is what you call the phenotype. So the problem is not phenotype. The problem is not crossing. The problem is the genotype. Because if you fail the genotype, you are most likely to lose the 3 marks. You can get 3 marks even if you don't know the genetic cross. As I told you that this is obtained from the question, check. You just copy here, paste. You get a full mark there. Then, because it is a man, I'm going to use X and Y. And because this is a female, is X, and please, we only use this X and Y because they said it is sex linked. If they have not said it, I repeat it because students make mistake. And when you're marking, you feel like crying and you have no choice. The only choice which is there is to cross. Yes, so X, but they said brown teeth, and brown teeth is caused by dominant allele. So remember, they have only one option. Then they are saying, yes, white teeth. Remember, white teeth can only be like this. So small b, small b. Now we give you another tick. Now don't forget this. You go to meiosis. Meiosis, what does it do? It splits these two and then splits that one also. So I always tell you that please don't leave them naked because they are not at the beach. It's only beach where they are people who are naked. Yes. And someone said that people at the beach are not naked. Okay, walk the way you walk at the beach on the street and then we see if you are not naked. Okay. Let's continue. Then they're saying that after that you have to do what you call fertilization. Fertilization. So fertilization here, you can use a Punnett square or you can cross. Let me just use a cross. The next time I will use a Punnett square. That it means that B, capital B, small b. This with this, capital B, yes, small b. Then this with this, xy and small b. Then this with that. Then we're going to have small b and y. And then these are called offsprings or the babies. And then this is F1. So when you're marking, how do we mark? We come here, we do this. We give you a tick. P1, F1, we give you a tick for that. You saying meiosis and fertilization, we give you a tick. Something I forgot. What is it? I forgot to say these are gametes. Please don't forget these steps. Please don't forget these steps. Because if you see now, you have 1 mark from the question, 2 marks me saying meiosis and fertilization, 3 marks you saying F1, P1. Even if you don't know the genetic cross, still you can get the mark. Think I'm clear? Yes. Let's continue. Let's continue. And then we say that, what was the question saying? Use the genetic cross to show the possible genotypic ratio. They want the genotypic ratio of their children. Now, I need to find out the genotype of this, the phenotype. They said genotypic is phenotypic. I need to find out the phenotype of these individuals. So, phenotype. Then I come here, say, this is a female who is normal. Female. You can't say female carrier. No. Why don't we say female carrier? Because we don't show. We can't see whether the person is a carrier or not. So it's going to be 2 female, female, yes, normal. Not normal. They specify that brown teeth. Brown teeth. Yes. Brown teeth. Yeah. Brown teeth. And then 2 male white teeth. 2 male white teeth. Yes. So if I've got those 2, what does it mean? They want the genotypic ratio 2 to 2. But if you leave it like that, we cross. You have to simplify it. Divide by 2, divide by 2, say 1 to 1. So that is the correct answer. Here we give you a tick. Yeah, I think I'm clear there. The question is, if you have any challenge with it, please let me know. Let me know. See if you have any challenge with this question. If you don't have any challenge, then we continue to the next question, because basically we're supposed to look at even a dihybrid cross. Supposed to look at the dihybrid cross. Okay, let's continue. If you have any question, please let me know in the comment. I'll try to answer it immediately. Kajibene United FC. I think now we are together. Yes. Let's go to the next. Ooh, yeah, it is a pedigree. Sorry, it's not a pedigree. It's a dihybrid cross. So this today's lesson is a mixture of a dihybrid and a pedigree. I also included the one question for blood group. Yes. So within 1 hour, the class will be done. So please take your time and then we try to discuss this question. These what? These questions you'll be able to uh, to 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 understand. All right. Humans, in humans, okay, and short fingers, capital F, and then widow's peak, capital H, are dominant over fingers, long fingers, and continuous hairline. Men, a man and a woman, both are heterozygous. They are heterozygous. The two characteristics, for the two characteristics, they plan on having a child. They are just planning to have a child. Okay, sharp. Let's go. Sex determination is caused by the 23rd pair. So we have chromosome number 1 to chromosome number 23, and these are pairs, meaning that they are equal to 46 chromosomes. But from chromosome number 1, yes, to chromosome number 22, we call them orosomes. From chromosome number 23, or the 23rd pair, we call it sex chromosome or gonozome. So when you're describing sex or sex determination, you're supposed to explain based on uh, based on the 23rd pair of chromosome. So we can say when they say show using a genetic cross, you're supposed to do all the procedure as I've been explaining to you. That is P1, F1. So if you are not going to forget, it's fine. You can leave the F1 for now until you reach the end. So now it's gonna be P1, which is phenotype. So what are you going to say here on the phenotype? You're gonna say male, then crossing with female. You see? Yes. And then they're saying that genotype. Genotype, you come here, you tell us that this is X, this is Y. Remember, males have have one X chromosome, and then they have one Y chromosome. So, and then this one is going to be XY. So, now what happens is meiosis. Meiosis is going to be XY. It's going to be XX, and these are gametes. So we circle them. Even if you don't circle them, still you get a tick. But when you circle them, it brings about that identity or separation. And then now fertilization occurs. This is X, this is X, this is XY, and then also this is XY. So basically that is it. And then now what happens? What happens is this is fertilization, and then these are offsprings. But don't forget that this is F1 offspring. So basically use gender. So it means that you have these are females, and then these are males. meaning that you have 50% chance of producing females and 50% chance of producing males. What is the question? Is genetic process to show the gender in humans and is determined by sex chromosomes of the parents. So in this case, that's what they want. If you check our booklet, the distinction material, and you go to genetics, you'll find out that this book is showing you exactly what I've talked about. What I've talked about. This is it. 22 chromosomes are called autosomes, and one pair is called sex chromosome. Males have XY, and female have. This is a description, yes, but what they want is this. If they say describe how sex is determined or done, then you can talk about this. But if they are not talking about that, then automatically you have to use because they said using the genetic cross. So basically they want this, they don't want the writing. Yes, let's go to the next question. Robin saying hi. Okay, yes, hi. All right. They're saying that the next question is what's a pedigree? Remember, in this pedigree, you have different styles. A question can come when this pedigree is sex linked. It can come when this pedigree is non-sex linked. So you have to take note of the following before you answer any question concerning about a pedigree. It can be sex linked, it can be non-sex linked, it can be caused by a dominant allele, or it can be caused by a recessive allele. So when you are trying to answer these questions, you have to first know this pedigree is caused by what? Is it a recessive allele? Is it a non-recessive allele, meaning that it's a dominant allele? Because if it is a dominant allele, then definitely you have to use the capital letters. If it is a recessive allele, then it means that you're supposed to use small letters. So please take note of that. Then if it is saying that, if the question comes and is saying that it is sex linked, if it is not saying that sex linked, please don't use X and Y. So we're going to look at these questions. We have different kinds of questions we're going to look at so that at least you don't suffer with this. Kundai is saying that I received a distinction. Thank you very much for receiving a distinction. And I know that at the end of the year, you will receive distinctions. I always say that distinction times two. All right. The first question is saying that these are national examination questions, like obtained from the past papers. Please always and always use these past papers so that they can guide you. All right, the first question is saying that moya is a disorder caused by a dominant allele. Moya is a disorder caused by a dominant allele, capital R. You see, the question is stating it that it is caused by a dominant allele. This disorder damages the arteries supplying the blood to the brain. Okay. So that is what is important here in this statement. We don't want to know supplying the blood. We don't want to know the condition. What is important is caused by a dominant allele. This is a key point. As I say that it can be caused by a recessive allele. It can be caused by a dominant allele. It can be sex linked, or it can be a non-sex linked, meaning that it is an autosomal character. So please take note of that. So in this case, first of all, it is caused by a dominant allele. And they are saying that they are not specifying anything here. They are not saying anything here that it is caused by sex linked. However much I've talked about male or female, I should not use X and Y. So let's go. The first question is saying that how many generations are represented in the diagram? When you talk about generations here, you only have to look at this is the first generation, this is the second generation, this is the third generation. So in other words, there are 3 generations which are indicated in this diagram. So the answer here should be 3. And then they're saying that give the letter, sometimes letters of an affected male. Now, sometimes we give you a key, which means you have to take note of it and then know that whether it is caused by a dominant, caused by a dominant allele, recessive allele, but we don't give you a key to show that this is a male, is affected, this is a female, is not affected. So we just give you the question. So please take note of that. So in this case, they gave you a key. So the one who is shaded is affected with the disorder. The one who is not shaded is not affected. So it means that you have to take note of that. So in this case, we are saying that affected male, a box. We say that a box means a male, and then a circle means a female. So if they're saying that an affected male, so here they're saying a box which is not shaded is unaffected. So they are saying that, so I come here and I find out it's only one person who is unaffected. So the answer is saying is supposed to be H. I think that's why it is also 1 mark. Then they are saying that genotype of individual A. Where is genotype of individual A? So because this is someone who is affected, yes, and this is not, they have not said that this is a sex linked. And then number 2, they produced a kid who is a female, and this kid is not affected. So what does it mean? It means that if this kid is not affected, it means that this person is recessive, small r. And then this one is not affected because this is caused by a dominant allele. So it can either be capital R, small r, or capital R, this individual A. So, but because this is small r, yes, and then they produce a kid who is small r, yes, it means that one allele came from the mother. Obvious, mother is like that, as we have seen it here, and one allele came from the father. Therefore, the father can't be this. So the father is like that, meaning that one allele came from the father, one allele came from the mother, and they gave rise to this kid. Hence, A is of this genotype. So it is A is heterozygous. I think I'm clear there. Yes. Then the next question, they are saying that genotype of A, we are done with that. It is capital R, small r. Give the letter or letters. Can you please start? I just logged in. Yes, we are going on. Yes, so if you have just missed it out, yeah, don't forget that we have a lot of questions down there. So try to keep up, and then you'll get there. So we are saying that give the letters of what? Let me check here. Someone is disturbing me. All right. Give the letters of individual who is not biological uh, kids for A and B. Where's A and B? A and B. So in this case, This line means they gave rise to these two kids. This person C and this person F, it shows that they just got married. You see, they just got married. So F and C are not biological children for this. So it means that the answer here is going to be F, yes, this F and C, because this, if this line was like this, then they would have been the kids of these parents. But because this line is not like that, it means that, what does it mean? It means that they just got married. So this is not a biological child for these two, and also this is not a biological child for these two. So I think I'm clear there. Then they're saying that, sorry, they're saying that brown enamel. This is another question. It's another question. Brown enamel of teeth is sex. Now here, this question, they have specified that this is a sex-linked. So in the question, if they don't specify that this is a sex-linked disorder, please do not talk about X and Y. However, you see now here, we have not talked about X and Y. So please find out whether it is caused by recessive allele, dominant allele, sex-linked, autosomes, meaning that non-sex-linked. So there is another question here. This question is about pedigree again. But this pedigree, what are they saying? One type of defect in human is carried on single allele. Okay. The diagram shows the inheritance of deafness in family. So they are not specific, that they are not saying that this is caused by is sex linked. No, they are not saying that. So if it is not showing you that is caused by sex linked, then automatically I have to keep it the way it is. Yeah, it means that you are not supposed to 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 check to use X and Y. Yes, you are not supposed to use X and Y. Just a moment. Okay, it's locked. All right. They are saying that how many generations? We have seen this question before. How many generations is one generation? This is the first generation. This is the second generation, and this is the third generation. So this pedigree diagram showing 3 generations. Children of Paul. How many children of Paul and Liz? This is Paul, and this is Liz. So the answer is gonna be 1, 2, 3. Yes, this is not a child of Paul and Liz. Why? Because this is just married. This means interbreeding or sex is happening, or marriage or cross, whatever you can describe it. And then yes, so it means that there are 3 individuals. So also it is 3. Then they're saying which phenotype is dominant. Which phenotype is dominant? Based on this, you have deafness, and then a person who can hear, deaf and hearing. You have to, since they didn't show you that this is dominant, this is recessive, you have to look at maybe 2 parents and then their kids, and then you see which one leads to the other. So if you look at this and this, this will not show you because we don't know whether it's heterozygous, or even this one will not show you. But this one can show you. This is showing this person is hearing, this person is hearing. But they produced a kid who is deaf. What does it mean? It means that these two, they had a recessive allele for deafness. It means that deafness is caused by recessive allele. Therefore, hearing is caused by a dominant allele. So, which phenotype? So the answer here must be hearing. Hearing. Yes. And then use the offsprings of Bob and Anna. Oh, Bob and Anna. So it's the same thing we have used. We use the same example. Why are you saying that hearing is the dominant allele? As I said that Bob and Anna, yes, use Bob and Anna to explain your answer. Bob and Anna, yes, look, we are seeing that Bob and Anna, both of them, if this is Bob and this is Anna or Anne, whatever, yes, if these two are normal and they intermarried or interbreed and they produced this kid, and this boy who is deaf, what does it mean? It means that this person, if it is small r, they are recessive. It means that Bob had is capital R, small r, even this one is capital R, small r, and then this came from the mother, this came from the father, and then they joined, and then they formed this kid who is, let me show you, a kid who is having the disease. It means that this allele, and this allele, they came here. Yes. So it means that, so use Bob and Anna and explain your answer. So you can say that Bob, who is a male and is hearing, yes, married, yes, Anna, who is female and is hearing. Did they say it's caused by sex linked? No. So that's why you are not saying anything with X and Y. So this Bob, who is hearing, meaning that Bob is capital R, small r, is heterozygous. In other words, heterozygous. Don't use small r, because they didn't give you the letters to use. Bob is heterozygous, and also Anna is heterozygous. They passed their recessive allele. Even Anna is like this. They passed their recessive alleles to one kid, and the kid became deaf, which is small r, small r. So that is the reason why Bob and Anna produced a kid who is having this defect of deafness. I think I'm clear. So sometimes they try to change it, tilt it, as long as you know the principle. How do you identify? Please look at the babies. Ne? If you see the babies, you'll see them that if one person is sick or is affected by the condition, it means that the parents are heterozygous. You understand? If the two parents are normal, it means that the two parents are heterozygous. But if all of them are normal, then it automatically shows that the parents are homozygous, dominant. Use letters A to represent a dominant allele and small a recessive allele. Show recessive allele to give all the possible genotypes for hearing individual. Possible. Use capital dominant allele and letter A for recessive allele to give all the possible genotypes for people who are hearing. It's 2 marks. Remember, we said it here. A person who is hearing, yes, and remember, is hearing is dominant. If it is capital R, say this, capital A is for hearing, ne? And then small a is for deaf. Yes, it means that a person who is hearing can be homozygous, like this, homozygous dominant, or can be heterozygous. This person is hearing and also this person is hearing. So these are the two options you can give. So we will give you a tick here and also give you a tick there. The last questions. I have this question and then I have another question and another question. So yeah, basically I have 1, 2, 3 questions to smash. Okay, let's smash these questions together. All right. They're saying that this question is now pedigree, but now this pedigree is sex linked. Is sex linked. You remember the question we saw? It was not pedigree, but it was sex linked. But now this one is pedigree and is sex linked. So we are bringing sex linked character in the pedigree. This syndrome, however, I don't know how you're gonna, whether it's hotty or gotty or whatever you read it, because it's an scientific term, but I know that many students read it as hot. Okay, is sex-linked genetic disorder? It is caused by a dominant allele. You see, it's caused by a dominant allele on X chromosome. This shows that this is a sex. I told you that you cannot use X and Y unless it's mentioned in the question. Yes. Or they're gonna ask you, is it sex-linked or non-sex-linked? The diagram below shows the inheritance of this syndrome in the family. Okay. Name the type of diagram represented above. We have been talking about it. Is a pedigree. Is a pedigree. Pedigree diagram. This is RNA. How many female are in this family? How many female are in this family? Remember, circle means a female. Yes, it's 1, it's 2, it's 3. What about this? This one is not a member. It just came to get married. Get married. Ne? But they're saying how many female? Okay. It's 1, it's 2. Since now they are going down, they're saying females, not kids. Yeah. Okay. It's 1, it's 2, it's 3, it's 4, it's 5, it's 6. Yeah. Yeah. They're not saying kids. They're saying all the females. So there are 6 females. Males in F1 generation. F1 generation. They're saying F1 generation. How many males in the first generation? So remember, you have first generation, second generation, then third generation. So how many generations are these? There are 3. So they are saying how many males in the generation? So it's only 1, the father. Yes. Then they are saying that the next question, they are saying that give Gabby's genotype. Who is Gabby? Ne? Who is Gabby? Gabby's genotype. Where is Gabby now? Oh, Gabby. Is it caused by a dominant allele? Yes, it's caused by a dominant allele. So if it is caused by a dominant allele, so what does it mean? It means that when you are writing it, yes, you're supposed to shaded means affected with age, because whoever is where is Gabby? Yes, this person is affected with this disorder and is caused by the. So it means that you have 2 options. Either you have Gabby is a female, so it can be capital G, small g, or it can be capital G, capital G, capital G. Yes. So, but the question is saying they give Gabizi genotype. So, which of the two is correct? Now you look at the babies. Is there any baby who is normal without the condition? Yes. Is this. And remember, this is the recessive allele. So it means that this is a male with small g, capital Y. And this is a female, small b, small g, small g. So it means that if this individual, if this individual is this one, if this one is a female, small g, né? And then this one is a male, small g, and then y. So the father is already this one. Yes, the father is already having small g, capital Y. Automatically, how did this individual and this individual get this C recessive allele if the mother is like this? Therefore, automatically the mother can't be this, so the mother is that, like that. So it means that this Gabi is, so the answer is gonna be X capital G, X small g, so that it shows that the individual is affected because of this capital G, but has a recessive allele which is being transferred to the offsprings so that the offsprings are free from the condition. And then they're saying that we are done with that. They're saying that Anju and Pilusa have 4 children. Give the phenotype of their sons. They want to know the phenotype of their sons, Anju and Pelusa, because they're saying sons. Okay. The sons. Okay. So now look. Pelusa is a male. Yes. Pelusa is a male. And this Pelusa is what? Is affected. So meaning that is having a capital G and a capital Y. But Anju is a female. Anju is what? Anju is a female, of which is caused by a recessive allele. You see, this is Anju. No. This is Pelusa. Pelusa. And then Anju is a female, meaning that it has no effect. And remember, it is recessive. So if they are looking for the sons, so how are these sons going to be? Automatically the son is going to have this. You know why? It's because the father will always give the come from there, will always give the Y to the son, and then the X is always coming from the mother. So the disease or whatever recessive conditions in males, if it is sex linked, is caused by the female. Why? Because the father is always giving the Y chromosome. So this came from the mother. And then since it is recessive, so the person is unaffected. So the description here, you're gonna say that, and for children, give the phenotype of their. So the answer is gonna be unaffected male, or unaffected, or these guys without Hortis syndrome. Yes. Explain your answer. So it's the same thing. This question is similar to the question we tried to attempt before. Explain your answer. Why are you saying that they're going to be like this? They're going to be unaffected. So you can say that Pilusa is affected. Yes, you see that Pilusa, we have seen it that Pilusa is affected. Yes, is a dominant allele. Yes. While Angie is unaffected. You can say unaffected or you can write this. Yes. Males inherit the Y chromosome, as I've talked about that males inherit the Y chromosome uh, from Pelusa. You see, these boys, the sons, they inherit the Y chromosome from Pelusa, and then they inherited this X with the recessive allele from Anju, and this Anju is already affected. Hence, the individual is going to be like that. So, how do I get the 4 marks? 4 marks by telling us what's happening to Pelusa and what's happening to Anju. Pelusa is affected, a tick, while Anju is unaffected. Yes. So the boys or the males will inherit the Y chromosome from the father. Give you another tick, yes, and then they will inherit the X with the recessive allele from the mother. That's all. Yes. You see, genetics is simple, but if you are not keen, it might crush you down. So you must always think before you try to answer questions concerning about this genetics. Please don't just rush and then you answer these questions. No, the questions you have to read them. You read them. You read them. You read them. You read them.
Welcome back to our class. Today we're gonna be looking at the paper 2023 final exam. Yeah, so share the video so that everyone is here. We can discuss this paper. No one should miss out. This paper was really a very easy paper for those people who did the study, and for those people who did use my book for distinction material. Yeah, if you want it, you always get it from the link below. So this paper was a little bit easy, was just a little bit of easy, and we're gonna see how we can really see that those people, they can mark themselves and then they see, and also those who want to prepare for the next exam. All right, let's start without wasting time. M.Saidi, as usual. According to Lamarck's theory, I know most of you don't like Lamarck because of his theory, but me, I like him because he started the story. All right, according to Lamarck's theory of evolution, or for evolution, acquired characteristics are not inherited. Obviously, we say that Lamarck says that acquired characteristics are inherited to the next generation. If you have a person, maybe your father gets an accident or is a bodybuilder, and then you're gonna produce kids who are having muscles already. Acquired characteristic is inherited. So here the key point is not. So you have to take note of that. So this one was off. And then they are saying that organisms evolve because they don't because they adapted to their environment. No, here they are saying that organisms evolve because of the adaptation to the environment. How do organisms evolve due to the adaptation of the environment? Is it natural selection? Because adaptation will require that those organisms which are able to adapt will evolve, and those organisms which cannot adapt will die. Yes. Then number 3, they're saying that there is variation among organisms. Lamarck doesn't talk about variation. Yes, so this one is off. Then they're saying environmental change has no influence on the species diversity. Here, they're saying that environmental change has no influence on the diversity of the species. And then here they're telling you that organisms evolve because they adapt to their environment. So how are these two different? How are these two different? Here it means that environmental change have no influence. For example, that if there is scarcity of food, yes, and then the one which is on ground die out. This is opposite of this, né? And they die out, and then there will be no influence on the species, how they diverse each they are different from each other. So that is wrong, because species will change based on the what they want, but still based on the environment. If they want to be, for example, they want to stretch their necks, they stretch their necks until they reach, for example, the tortoise. If we are using the what, the giraffe, yeah, you call them giraffe, yes, because that's the best example most of the people are looking at. So the best answer here is gonna be B. And then they're saying that genotype for species characteristics. When you talk about the genotype for species characteristics, what does it mean? They're saying that contain 2 chromosomes. No. Physical appearance, they're saying genotype. Physical appearance is a phenotype. No. Is the composition of a gene pair. Look, they're not saying that genes. They're saying gene pair, meaning that you have. Remember that when you talk about an allele, is also a gene, but is half of a gene or alternative form in which a gene can exist. For example, tallness is an allele, and also shortness is an allele. For example, capital T, small t, for example. Yes, so this is an allele, this is an allele, but this is a gene, meaning that it's a pair which makes a what? A gene. And then is represented by the genotype of a specific characteristic. Here, characteristic. Here they're saying it is represented by one allele. It's not represented by one allele. Even if it is shortness, it will be represented by the 2 alleles. Yes, for example, capital R, or small t, they are saying a characteristic. Yes. So when you talk about the characteristic, it means that they are looking for the complete set of that characteristic. So the best answer here becomes C. And then they're saying after discovery of fossil, scientists classify it as after discovery of a fossil, scientists classified it as an African ape because it had, okay, what do African apes have? Large cranium? No. Small cranium? Yes, they have small cranium. Small cranium? Yes. Large cranium? No. So meaning that A and D are out. So these 2 are out. So let's look at this. Small cranium, non-prognanthus, meaning that they don't have a protruding jaw. Apes have protruding jaws. Yes, prognanthus means protruding jaws. So prognanthus, this is correct, this is wrong. So it means that the best answer is C. They're saying that large canine. This one is correct, this one is correct, but this is wrong. Therefore, B is wiped out. The best answer is C. So C is going to be the most correct answer. Yes, let's go to 1.1.4. The statement below describes the step in the process of cloning. I explained this, the process of cloning yesterday very well, but let's ignore that you are not there. So now let's look at it. Remember, they are saying that embryo. Okay, let me just show you here, so that number 1, you get one cell, you get a cell A and cell B. So this is animal A, animal B. Get a cell, ovum. I'm just summarizing it very fast. Somatic cell. So here is somatic cell, meaning that is 2N. After that, remove the nucleus of the ovum. Yes, throw it away. Take the what? The casing. And then here, remove the nucleus because I want the characteristic of this. Bring the nucleus, throw the casing. So bring the casing here and bring the nucleus, fuse them together with electric shock. Yes. After that, bring these 2 once it is fused together, it looks like a zygote-like structure. Therefore, it becomes, I'm just explaining so that because this is the process they are trying to show you, it undergoes mitosis. Bas. Then, let's go back to the question. They're saying that the statement below describe the steps involving in the process of cloning. Embryo in the implanted is implanted into the uterus of an adult female. Embryo is implanted. This must be around this stage 4. Okay, we shall find out. Nucleus from the somatic cell of the donor cell is extracted. This could be 1. Nucleus. Check. Nucleus. They're saying the nucleus or the cell? Nucleus from the somatic cell. That's not step 1. You can't extract the cell before you get the cell. You can't extract the nucleus from the cell before you get the cell. So we need to first get the cell. We are looking for these 2 steps of getting these 2 cells. All right, we go back. They're saying that the nucleus from the somatic cell is inserted into the ovum. The nucleus from the ovum of another is removed. The ovum from, okay, now we have to look for it. They're saying that the nucleus from the somatic cell, meaning that, okay, they obtain the cell, yes, but now after obtaining the cell, in this statement, they're not talking about this. So meaning that they're starting from this. They're not talking about this. They're starting from 1, 2. Okay, let's go back. They are saying that the nucleus from the somatic cell of the donor is extracted, meaning that the nucleus is removed, is brought here. So this could be step 1. Okay, we shall see if it is the most correct. Step 1, so this is covered. They are saying that the nucleus from, the nucleus from the somatic cell is inserted into the ovum. The nucleus, this is done. The nucleus is of the somatic cell is inserted into the ovum. Do we have the ovum? The nucleus from the ovum of another individual is removed. This is step 2. Yes, this should be step 2. Because I removed here the nucleus, so I remained with this. So now I inserted this into this. This should be step 3. The nucleus from the ovum of another cell is removed. It's done. The ovum of the new cell is given. The nucleus from the somatic cell is inserted. Now this is step 3. So meaning that now I have 2, 4, 3. Then I'm remaining with this and this. So they are saying that the ovum is now, the ovum with a new nucleus is now given electric shock. Now we are here. We are giving it electric shock, meaning that now this becomes 4. Yes, and then now the embryo is implanted into, so meaning that I'm going to have, yes, it's going to be 2, yes, 4, yes, 3, 4, 1, meaning that 2, 4, 3, 5, 1. So this is the most correct answer. So the answer is A. Let's go to the next question. Then they are saying that 1.5, shrubs, these are short grasses, of the family. This family, they are saying that can be found in Australia, South America, Indochina, and parts of Africa, as shown in the map below. It is hypothesized that all continents were once one large continent called Pangaea, and that they separated due to continental drift. They said, okay, sharp. They're saying that this is evident. This evidence that the family, yes, this family, what about it? All belong to the same species. No, they don't belong to the same species. They don't belong. That's not an evidence to show that they belong to the same species. Number 2. They're saying that all they are equal distributed in all continents. Who told you that? They're not equally distributed. Are you trying to mean that what is here and what is in South America and what is in Africa, they're equal? No. Wrong. They're saying become extinct. They're still existing. Arose from a common ancestor. Simple, because at first they were together, yes, but they went to different environment due to the separation of the continent. And then they developed into different species. That's why we say that they don't belong to the same species. Therefore, D is the most correct answer. 1.6. Yeah, these ones, they take some time, but when you reach there, we're going to be faster. The diagram below shows the DNA profile of a girl that her mother and 4 males are about the males. Okay, maybe they are claiming. Which male is a girl's biological father? I have to look for all the bands which correspond to the mother, because the mother is known. This goes with the mother. Done. This goes with the mother. This goes with the mother. Now, the remaining bands from the girl must be matching with the guy who is claiming. Okay, let's go. This one matches. Cool. Yes. This one matches. Is there any? Yeah, because this one is matching. So is there any other for the girl? No. So it means that this is number 3 is the biological father. All of them must be ticked for the case of the girl, but they must be the remaining one which did not match with the mother must match with the father. Hence, number 3 is the most correct answer, which is C. Okay, the following are the characteristics of group of animals. Yes, animals. If you're saying animals, just a moment, guys. Okay. Just a moment. Okay. Animals. They're saying that which characteristics of group of animal which are the characteristics of group of animals able to interbreed, occupy the same habitat, produce infertile, belong to the same species? Which combination correctly represents a population? Which combination represents a population? Yeah, it means that you can't know which group represents a population. Just a moment, guys. Let me increase here so that I can see clearly. Yes. Which population does the population is being shown by what? Is being shown by the able to interbreed, and they must produce a fertile offspring, occupy the same habitat. Yes, they must be able to interbreed, but interbreeding they must produce the fertile offspring. And the only word which can help us to bring about to produce a fertile offspring is one word. Which word is that? Same species. They belong to the same species. So the answer is B. Same habitat, same species. Yeah. So we go to the answer becomes 1, 2, 4. 1, 2, 4. Where is 1, 2, 4? Is there any 1, 2, 4? 1. 1, 2, 4. Yeah, it's B. Yes, it's B. So yeah, it gives me the correct answer. Let's go 1.8. Which diagram? Let me see. Which diagram? Not which diagram. During anaphase 2 of meiosis, the 2 chromatids of the chromosomes are pulled apart, each moving towards the opposite poles at a rate of 1 micrometer per second. This question? Okay. I see it as a new content to students. Anyway, but it's just an application question. Okay, they're saying that, yes, 2 chromatids of the chromosomes are pulled apart, each moving towards opposite poles at a rate, this is the key point, at a rate of 1 micrometer per second. Okay. Each second, 1 second, it means a distance moved is 1 micrometer. Let me say 1 like that. The distance in micrometers between the chromatids after 20 seconds. Obviously, if it's 1 second, 1 micrometer, after 20 seconds, they'll have moved 20 micrometers. So that is a direct question. However much is just an application question. So the answer becomes B. Yes. So the answer there becomes B. Yes, let's go to the next question. The next question, they're saying that, which one of the following indicates the type of variation? I gave you examples yesterday. Anything which does not change since you are born is continuous variation. Anything which changes as you are growing up, it's continuous variation. For example, here they give you height. Height changes, therefore is continuous. The way you were born and the way how you are now is not the same way. Height is continuous. So is where there is this continuous is, I think I have the answer now. Because the rest are wrong for the height. And it can't be correct for the next one. Okay, skin color. I don't think most of us, we are born yellow bones. Check us now. Hey, we are original indigenous. Yeah. Yes, color changes, skin color changes. You check the kind of people. Some people, I want the black one, I want the light skinned one, I don't know what you're gonna use for them for, but it changes among the individual. All right, this is continuous. Check. Earlobes, the way those earlobes are since we are born, unless you get an accident and then they change, but if not, it's the same thing. So correct. So let's not waste time there. Answer is B. Let's go to terminologies. I know sometimes the terminologies, they make these students a little bit of a problem with it, but I don't think these terminologies were that hard. Ne? Let's go. The first terminology, they are saying, the type of inheritance which produce intermediate phenotype. Intermediate phenotype. Don't forget that we said it in the what? In the distinction material book, our distinction material book. Ne? Yeah, for those people who were not there. But okay, there we go. Intermediate. How do you call this? Incomplete. Incomplete dominance. Intermediate. Yes, we talked about it. So the answer here is incomplete. Complete dominancy. Yes, done. The point at which adjacent chromatids overlap during crossing over. Chiasma. Guys, spelling is mandatory. The process by which information is copied from DNA to RNA, transcription. Told you. Transcription. Let's go to the next question. What are they saying? They're saying that sugar found in DNA, sugar, thymine. They're saying sugar, not base. Gone. Thymine, that's a base. They're saying sugar found in DNA. Ribose is found in RNA. So for the DNA, it's going to be deoxy. That's why he's saying D. DNA, deoxyribose nucleic acid. So the sugar found there is deoxyribose. The bond that occur between nitrogenous bases in DNA molecule. The bond that occur between nitrogenous bases in DNA molecule. So how do you call that bond? That bond is weak hydrogen bond. Yes, you must say weak hydrogen bonds. Most of the people, they wrote peptide bond. Peptide bond. In some cultures, they say that sometimes you can laugh because of sadness. Not because you are happy, but you are sad. The same way someone can cry because of happiness. When I laugh at because someone is because of the sadness. So this is weak hydrogen bond, not peptide bond. Yes, because I highlighted it that these are the mistakes students do. Yes, okay, let's go. Manipulation of in organism's gene to obtain the desirable characteristic. How do you call that? Manipulation of a gene to get a desirable characteristic. Whenever you talk about manipulating in science, whenever you say manipulating, what did I say? I said that is engineering. Engineering, manipulating. But you are manipulating genes. So, what are you manipulating? Genes, therefore, genetic. Simple. Genetic engineering. Guys, I told you, you don't need to cram. You don't need to cram. You just understand life science. Tell the people to join the channel. If you want a distinction, Thunder a Duke. Yeah. Use the distinction material. There is no way how you can fail a paper. Never. Yes. Okay. Unless you don't want to. All right. Oh, unless you found me. Or you found the channel late. But if you want a distinction, tell the whole world. Thunder a Duke. Okay. They are saying that representation showing the arrangement of diploid set of chromosomes. Hey. Representation. They are looking for how do we call that representation? Which shows the diploid set of chromosomes, meaning that chromosome number 1, chromosome number 2, chromosome number, but they are saying, see, diploid. Like that, we call it a karyotype. Karyotype. Done. Terminologies, out of 7, 7. Okay, source of variation. How do you call random? We talked about this yesterday. Random mating, random fertilization. Source of variation for the species. I told you that in fact a man produced over 500 million sperms. That is a rich man. That's why they tell you that you are 1 in a million. It means that your sperm was 1 in a million sperms which were released by your father and 2 there, and then only 1 you fertilize the ovum. So random fertilization is also random mating. You don't just find people come and say that, okay, now I want this one. You just go and then marry. You should just say, I want the small one, I want the big one, I want the light skin, I want the dark skin. You see, you're not doing it randomly. So you are minimizing variation. So variation, it must be random mating. When you are chasing this one and then another one comes into your eyes, you just go with that random mating, like a chicken. Yes. So this is both A and B. The answer is not A and B. The answer is both A and B. I explained it yesterday. Both A and B. Organisms that possess 2 factors which separate so that each gamete contain only one of these factors. Law of dominance. There is no domination here. Law of independent assortment. Organism possess 2 factors that which separate that each gamete. Law of independent assortment. This is done in dihybrid cross. We call this law of segregation. Law of segregation, not principle of independence. So the answer here is none. Because this is a law of segregation by Mendel. You remember that guy Mendel discovered the law of segregation in he was there in 1865. Yes, Mendel, Georg Mendel, that priest. Yes, Georg Mendel. Yes. Guys, life science is nice. So what you should do is be nice to it. Once you are nice to it, the subject is gonna be nice to you. If you are not nice to it, forget about. Okay, guys, just 1 second so that yes, Mr. Ramaphosa has given back electricity to us, so now we are happy. Yes, it's back. Yeah. All right. So if you see some disconnection a bit, just know that you're just switching the system. But we are continuing. Reproductive isolation. Reproductive isolation. Is my mouse. Okay. Reproductive isolating mechanism in species. You separate organisms through reproductive isolation, breeding in different seasons of the year. Correct. Infertile offspring. This is not correct. This is not correct, Baba. Look, same. This word, these people are confusing us, ne? Breeding in the same season of, no. It's supposed to be breeding at different seasons of the year. Let me show you what these people are trying to trick you up, ne? Let me just show you using the distinction material so that you see exactly how these people are trying to this is a real fake. They are faking us, so that we are off. We are sober. All right. Let me show you what they are showing you. Breeding, yeah, is here. You see, breeding at different times, not breeding at the same. So that answer is wrong. So this answer is wrong. You see, when you're saying the same, the answer is supposed to be breeding. This distinction material, there is no way you can go astray. Yes, so this is wrong. Infertile offspring. Correct. Check here in the book. Check here. Infertile of the way it is without even changing anything. Yes, infertile offspring. So you see, we say that mechanism of reproductive isolation. Now there are some people saying that I wish I knew this booklet. Sorry. Ne? Yeah. But you can refer to someone else so that you can get rewards. All right. So the answer here is B only. Yeah. Let's go to the next question. What does it say? They're saying that this is a phase. Ne? What I've seen in this paper, ne, as I'm going, ne, I don't know that much what is down, but what I've seen it here is they are using small words to take you off. If you don't read these questions very well, they're just using very small words just to take you off. All right, let's go. So we have to be keen, guys, when we are trying to answer. Yeah. So even may have to read too much so that I don't go astray. I might go astray and give you a wrong answer. No, man. All right, let's go. They're saying that the diagram below represents a cell in early stage. You see these people. The word early. That word, there is early and late stage, mid stage, early stage. So now they're saying early stage. Okay. So if you look at this, which stage is this? This is not interphase. This is early prophase. Chromosomes which are homologous, they align together. Homologous. Yes, crossing over is not yet there, but they have aligned together. And you see that these dots shows that the nuclear membrane is starting to disappear. The nucleolus is already disappeared. You see? These people. So this is prophase. Prophase. Which prophase? Prophase 1. If you don't say prophase 1 is 0. Number of chromatids. So how many chromosomes do you have? 1, 2, 3, 4, 5, 6. So we have 6 chromosomes, but each chromosome has 2 chromatids. So 6 times 2, which is 12. So the answer here is going to be 12. Number of homologous pairs. They are saying the number of homologous pairs. They're not saying number of chromosomes. Pairs. So meaning that 1 pair, 2 pairs, 3 pairs. 3 pairs. Someone is going to write 6. Ask you, do you have eyes? Yeah. Do you see 6 there? No. I see 3. Why did you write 6? I don't know. Okay, let's go to the next question. Identify what is R. Yeah. R is the nuclear membrane. Nuclear membrane. What is S? S is cell membrane. Membrane. Yeah, you know, I told you last time that you guys, you confuse nuclear membrane and the cell membrane. T. What is T? What is now T? T is nucleolus. Nucleoplasm, not nucleolus. Nucleoplasm. So, the nuclear, okay, maybe they will slash it with the nucleus. Nucleus, yes. But based on this is nucleoplasm or nucleus. So if you gave nucleoplasm, Yeah, they might give you. If you gave nucleus, they might give you based on what they have shown us. Ne? Yeah, don't say that. No, me, I wrote nucleus. Yeah, you might be right. Ne? Yeah, correct. All right, let's go. 2 organelles in animal where meiosis takes place. They're asking your parts, the most important structure on you. Which structure is that? Testes. Testes. That's one. Oh, if you wrote it like this is also correct because this is singular, this is plural. Okay, I'm not saying that you write 2. No, just write 1. Hey, students, that's number 1. Number 2 is ovary. So they give you a tick here, they give you a tick there. Simple. They're saying that, they write these funny words of these plants. Okay. Who knows this? I don't know. Let me just quote. The plant carries edible fruit. Scientists have been studying the inheritance of these 2 genes. One for the stem texture, and then another one is for the fruit shape. Okay, sharp. Let's go. The thing, stem can be okay, smooth. Smooth is represented by a capital letter, and then prinkly. Prinkly means that they have those yes, small letter, and then the shape round is capital letter, elongated is small letter. The type of cross, dihybrid. Dihybrid. Dihybrid cross. They're saying state the dominant characteristic in the stem texture. Texture is here. Ne? Smooth. Answer here is smooth. Smooth. Boom. Done. Let's go. Recessive. Recessive for what? Fruit shape. Fruit shape. Elongated. Let's go. 1.5.3. Give there. The genotype. Their genotype. It means that you are supposed to write in terms of gene. Prinkly, stem, and elongated. Prinkly is small letter, so I can't be with the heterozygous. Small n, and then elongated. Elongated is small r. Listen carefully. If you wrote NNRR is wrong. Ask me what? Why? Because of this space. I explained it yesterday. Yes, a space must not be there because this is a genotype. These are not sperms. It's a genotype. So it's supposed to be 1. Yes. They're saying that phenotype of plant with capital N is what? Because the small n would be masked. Capital N is smooth. And what? Capital R is what? Round. Smooth and round. Sharp. If you have the space there is cool, but the genotype is supposed to be no space. Yeah, let me know if you have questions. Ne? If you have questions, if you have challenges, yes, I'll smash them. Yeah, I'll smash them. Yes. Okay, let's go. Let's continue to the next question. Which question? Alright, they're saying that the table below shows the information selected on hominid fossils. Okay. Alright, we are hearing. What are they saying? What are they telling us? They're saying that P. What is P now? P. Oh, the common name. Australopithecus sediba carabo. We say this is a transitional fossil. Missing link between Homo habilis and the okay. Who discovered did a bagger? I don't know why they like carabo too much. They brought it in 2017. Yeah. It was the second or third objective. Yeah. Is read the bagger. Lee. Beggar. Yeah. Like that. This is Sediba, Carabo. Yeah, you can and you can find that someone called Carabo is feeding this Carabo. All right. They're saying that Taum child. Oh, Taum. If you're coming from northwest. Yes. How do we call? Remember, I told you don't go the paper without knowing the 3 Australopithecus africanus species. I said Taum child or Taum child, little foot, Mrs. Place. What are they looking for here? They're saying this is Australopithecus africanus. africanus. Cool. Just saying A. africanus, they would have given a tick. Yes, because there is no Ardipithecus here. So tick A. africanus. But they have already given you the spelling here, so there is no stress there. They're saying that who discovered? Who discovered the town child? Raymond that. Yeah, it's Raymond that. Raymond. I don't know whether you call it Raymond or Raymond. Raymond. That. Raymond that. Check that booklet, the distinction material. Check this distinction material. I organized it nicely for you guys, and even I created a video. Born. Check. Australopithecus, Carabo, read the bagger, Malapa. And then they're looking for the 3 Australopithecus africanus species. Check. Born. Raymond that. Taum. Sharp. Let's go. They're saying, name the fossil. Name the fossil. P. We have said it. P is Carabo. Ne? Then they're saying, identify species R. Species R. We said Australopithecus africanus. Let's go. They're saying that, give the name of the scientist who discovered Q. We say that, read the bagger. Yes. Well, you can also give that one brief Ilof. Yes, Ilof. Where is he? Yeah, this one. So it depends on whom did you give. Yes. Then they're saying S, we say that it's Raymond that. Now, by this time, you have 50 marks out of 150. This one is enough for you to shift from a failure, meaning that you're above 30 now, 30%. However, it's not enough for my students like you. Guys, don't have too much pressure. Like, now I'm failing. That's life. Still doesn't mean that if you fail this, then life has stopped. No. All right, let's continue. It just make you to prepare, and I pray for you guys so that you pass this, so that you get enough gelete. All right, let's go. They're saying that X, what is X? That is amino acid. I told you guys that you must always label these things before you start. Amino acid. Simple. Let's go. X, Y. What is Y? Y is anticodon. Z, messenger RNA. Let's go. X, amino acid. Z, messenger RNA. Give the nitrogenous base of sequence, of DNA base triplet, complementary to complementary DNA base triplet, complementary to mid codon of Z. Oh, they're saying DNA, meaning that we have to convert it backward. Let's say this is 3, this is 3, this is 3. So we are looking for this. So, but we are converting it backward. You have A, U, G. So we're going to be, say A goes with T, U goes with A, G goes with C. So this is the answer. That's what they are looking for, because they're saying mid in the middle one and for DNA. Then they're saying that give the nitrogenous base of Y. Where is Y now? Oh, the anticodon. So meaning that they want you to change this into transfer RNA, meaning that A goes with the, remember A transfer is an RNA. RNA doesn't have T. Therefore, we're gonna say U. U goes with A, and then G goes with the C. So the answer is this for this question and this for that question. Yes. Describe the process shown in the diagram above. Hey, what is this? This is translation. I told you guys. How many marks are this? Guys, don't tell me that you disappointed me unless you are not trusting me. Don't tell me that you disappointed me. There is someone who told me in the comment, I was so sad, but I didn't stop. He told me that you lost me in the first 5 minutes. I knew that this one is looking for attention. Yeah, because I talk too much. Okay, I talk too much, but I wanted you to understand the concept. Yes. Anyway, what do we mark? We can have different ways how we can mark. But this one you are describing, name and describe. The process is called translation. That's one mark for the word naming. And then according to the messenger RNA, the transfer RNA with the complementary anticodon bring the required amino acid to the ribosome. The amino acid are joined together by the peptide bond to form a required protein. Simple. That's how I have been describing. But still in my book, the distinction material, I showed you exactly what you are supposed to do. Now the mouse is jamming. Okay, doesn't want me to this mouse doesn't want me to give you okay, let me use this. Okay, this mouse doesn't want me to give you answers, ne? Sorry. Yes, born is there. Each transfer A is a marking point. Here is a marking point. This is a marking point. Each transfer A carries a specific amino acid. We give you a tick there. When anticodon on the messenger matches the codon on transfer A, we give you a tick. Specific amino acid, we give you a tick. Matches the anticodon. Yes. Then we are saying that this brings the required amino acid. Yes. Where does it go? To the ribosome. The amino acids are joined together or attached by peptide bond. Guys, we call them peptide bonds. We don't call them polypeptide bond. We call them peptide bond. I was very clear when I was explaining this. And then to form a required protein. That simple. Short as that. But you can find a person writing a full page. So you're okay. That's short as that. That's what we wanted. Let's go. So this description thing, I'm not going to waste too much time because it is open thing, né? And you can find them in any book. Yes, you can find them in any book. Yes. Let's go to the next question. What are they saying? Just a moment. Yes. What are they saying? They're saying that the table below shows the codons. I explained this. Guys, take note of this because I explained it and I said this is one of the confusing subtopics. The table below shows the codons of codons that code for some amino acids. Okay, sharp. A mutation caused by DNA triplet to change this. Okay, so it means that we changed this to this. But they're saying this is DNA. But what is here is messenger. Meaning that to read this, I have to change it first. So I'm going to say that, okay, this was C. C goes with G. T goes with A. And then G goes with C. But change the two. C goes with G. T goes with A. And A goes with U. Because this is messenger RNA. Wabona? Yes. Now I come. I go to, you go to what? I go to this table. I look for it. GSC. 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 GSC is aspartic acid. What about this? Does it correspond to a different amino acid or the same amino acid? Let's go. And then you're saying that GAU, aspartic acid again. Wabona? Ntuli. I can call you Ntuli. Yes? Isn't it? I'm just saying Intuli because Intuli called me and said that, thank you very much, sir, for teaching me. Yeah, I think I managed to pass. I pray for you guys, so that you pass. Sometimes when you're marking you're marking your students, and then you get something, you think of something, you end up giving a wrong one correct. You never know. Make sure that you pray until the results come back. Yes. Anyway, so since they code for the same amino acid, yes, so it means that the protein will not change. But these are 4 marks. Where am I going to get the 4 marks? You're gonna say that this change of this, yes, this changed into this. You writing it GAC changed to GAU. Those are marks. Those are 2 marks. This changed to this. Why this one is a mark and this is a mark? Is because you changed it. You see, you changed this. That's 1 mark, and you changed this. Those are 2 marks. And then this code for the same. This code is for aspartic acid as well as this code for aspartic acid. Meaning that these two, they code for the same amino acid. Is another tick. Those are 3 marks. Then the 3rd mark, the 4th mark would say that this will not change the protein formed, or it will not change the functioning of the protein formed. Simple. Why? Because it's like calling you with the first name, calling you with the second name. You are the same person. Yes. Or sleeping on your bed when you are straight, and sleeping on your bed when you have carved somewhere, or does not mean that you're a different person. It's the same person. So they're just saying that, yes, however much the mutation occurred, but the mutation did not change the amino acid. Simple. This question, they have brought it back again. Tabulate 2 difference. I emphasized this question of mutation, and I told you that I know what students find when it comes to this question of mutation. Anyway, this question. Tabulate 2 differences between DNA nucleotide and RNA. I explained it before. Yes, those people who were on my channel yesterday. They are they are not asking you the differences between DNA and RNA. No. They are saying DNA nucleotide and RNA nucleotide. Clear? So there are 2 differences. The first one, DNA nucleotide, has thymine, and the RNA nucleotide has uracil. The second one, DNA is made up of, nucleotide is made up of deoxyribose sugar, and RNA nucleotide is made up of ribose sugar. There is no any other difference. They are looking for the nucleotide. They are not looking for DNA. They are not looking for they are not looking for just those nucleotide for the RNA and for DNA. That's all. So if you wrote a lot of things there, but there are 5 marks. Where is the 4th mark? 4th mark is for the table. Just drawing a table and say DNA nucleotide, nucleotide. Even if you didn't write anything here, you draw a table, a complete table, you will get a mark. At least you are not getting 0. Yeah, at least. Yes. Let's go. Down syndrome. I talked about it also. Down syndrome. Okay, let's go to Down syndrome. What are they saying about Down syndrome? Okay, let's go. Down syndrome is the result of individual having extra chromosome on chromosome number 21. Okay. The genetic variation that can be caused by Down syndrome are, okay, trisomy 21, all somatic cells individual, 3 copies of, okay, chromosome number, all. This word is a key point. And then they're saying that mosaic means something which is moving. Yeah. Okay. Individuals have only some cells, not all. So this is a key point versus this extra chromosome, which is caused by abnormal process of cell division after. This is after, is caused by after fertilization, but what occurs during gamete formation. This is during gamete formation. This is after fertilization. You see? Yeah. That's the major difference. The type of mutation lead to trisomy 21. The type of mutation that leads. You can say chromosomal aberration. Those who wrote chromosomal aberration, correct? And then chromosomal mutation. Because these are chromosomes. Chromosomal mutation. Yes. Or aberration, correct. Abnormal process during gamete production that lead to 3 copies of abnormal process that lead, name the abnormal process that lead to the 3 copies of non-disjunction. The type of cell division that occurs after fertilization, it is mitosis. The saying, describe how the process mentioned above, which above B here, A, non-disjunction lead to trisomy 21, is 5 marks. This is a general question. Yes, go back to my distinction material so that we don't waste too much time there. Yes, I talked about it, but sometimes people, they have challenge. But before I left meiosis, I said that please don't go without knowing this process, because this process, they will ask you. Here is a question. Non-disjunction, failure of the chromosome to separate is they are looking for this. You see, this page. Each nucleus has it contains 23 chromosomes after meiosis, but non-disjunction when occurs, okay, one nucleus will contain 22 while the other will contain 24. Yes, either of the resulting gamete will fuse with the normal gamete, but they said yes, with the normal gamete. They said yes, they said what will happen. Yes, so the normal gamete is 23, then the resulting the 20, if the 24 fuses with 23, it's gonna be 47. The 22 fuses with the 23 is gonna be 45. Yes, so in this case they are looking for 23 because they're saying trisomy 21. It means that the 21, it has an extra chromosome, not short of a chromosome. Yes, let me explain to you. Yes, and then they are saying that let me show you what they are trying to say here. Yes, and remember, during non-disjunction, this is a cell under metaphase. Yes, so this is what happens. So during non-disjunction, the whole set is going to go to one side, so meaning that I'm going to have 2. So the normal one is going to be 2 cells. I'm going to have 2 here and 2 here. But during non-disjunction, the whole of it is pulled to one side. And then this one, yes, it will go to one side. And then this one, another one will go to one side. Meaning that here, yes, here I will have 1, 2, 1, 2, plus this one, 3. And then this side, I'm going to have only 1. So this one has extra chromosome. 2, 3 instead of 2. And then here it has short. So they are looking for this because they are saying yes, if it occurs on chromosome number 21, it becomes trisomy 21. Tri means 3. 1, 2, 3. You understand? So now when this one fertilizes a normal ovum, then it's going to have, now is the cell is supposed to have 23 chromosome. But this one is not going to have 23 chromosome. It's going to have 24. So if this fertilizes a normal ovum, which is 23, it's going to result in 47 instead of 46. So that's what they are saying here. So go to the distinction material. Yes, get there. So that's what they are trying to explain, or they're asking you here in this question. Let's go to the next question so that, yes, this describe 2 differences between trisomy 21 and the mosaic Down syndrome. Yeah, I think I talked about it. Trisomy 21, all cell, check, all somatic cells in the individual have 3 copies of trisomy of chromosome 21, you see, all. But here, yes, make sure you draw a table. I think they don't want a table. They're not saying tabulate. While here in mosaic, the individual has only some cells with extra chromosome 21, but here all the somatic cells. The number 2, the process, when does it occur? It occurs during gamete formation, while here it occurs during or after fertilization. Those are the 2 differences they are looking for. It's just as something from there which you can use to get the answer. All right, I see ourselves trying to finish soon. Yeah, so we are going to the question number 3. Question number 3. How is the paper? Let me know. How is the paper? Let's go to the next thing. Question. Yeah, I think the paper was simple. The paper was simple for those who read. Yeah, let me say like that. And I think I should come back and I teach you how to read. Does not stop only here, even when you go to the university. Good. All right. The diagram below shows the inheritance of tithe. They like this funny name. Anyway, I'm not going to remove my teeth because of these names. Okay. Is a rare disease which lead to this destruction of the neuron. It is inherited as a autosomal. Okay, autosomal means that it occurs in the body cells, meaning that it's non-sex linked. It's occurring in these other cells, these other chromosomes, not on chromosome number 23. Yes. Okay, TT. So meaning that you must not use X and Y. This is not sex linked. It's autosomal disorder. Anyway, let's go. Describe what is meant by is a disorder which is caused by a gene carried on non-sex chromosomes. Yes, non-sex chromosomes, meaning that from chromosome pair number 1 to chromosome pair number 22, it must not be sex. Yes. How many sons do individual 1 and 2 have? 1 and 2. Yes, sons. Son is a box. It's 1, is 2. But this son is not belonging to these 2. You understand? This guy, yes, just married the daughter of these 2 guys, meaning that the answer is 1. Understand? You see? Yes, this is just an intruder who came to marry the daughter. Yeah, the beautiful daughter of 1 and 2. Yes. So the answer here is 1. Using the individual 3, 4, 5, explain why it can be concluded that this disorder is controlled by a recessive allele. 1 is 3, 4, 7. 3, 4, 7. Okay, let me clear up here so that at least yes. Okay, let's see. 3. Okay, this is the affected individual, and these 2 people who are, if they produce a person who is affected, it means that these 2 people were heterozygous, meaning that it is capital T, small t, capital T, small t. You see, since they carried that since they carried that gene, this gene, recessive allele, and then this recessive allele and this recessive allele gave rise to this, meaning that, what is the question? What are they saying? Which question is this? 2.5.3. They're saying that it means that individual 3 and individual 4, they are both normal individuals. I see. That's the first mark. But they gave rise to individual 7 who is affected with the disease. You have to tell us the disease, a female who is affected with the disease. Yes, that's the second mark. This means that individual 3 and 4, they were heterozygous, meaning that they are heterozygous because they are both normal, but they gave someone who is sick, meaning that they were heterozygous, they were carriers. That's another mark. And then they pass their recessive allele. one recessive allele from 3 and one recessive allele from 4 to individual 7. That's another mark. You're able to get all the 5 marks. One recessive allele from 3, 1 mark. One recessive allele from 4, another mark. Yes, to form this one who is affected with the disease. So yeah, it is an application question whereby you have to apply and explain exactly what happened to inform individual 7. Then they are saying that individual 1 and 2 can produce children with 3 possible genotypes. Okay, list those 3. Individual 1 and 2 can produce, yes, individual 1. 1 and 2 is the same thing. 3 and 4, they are both heterozygous. Okay, they can produce. If they are both heterozygous, it means that, look, it means that this is capital T, small t, heterozygous, capital T, small t. So it means that when they produce, they're gonna produce capital T, capital T. This is what they are trying to show you. Capital T, small t. This is capital T, small t, and then small t, small t. You check these two. They are both heterozygous. This is homozygous and this is a homozygous. Let's go. The question. They're saying that list all the possible genotypes that have 25% chance of offspring. Then this is 50 and this is 25. This is 25. Meaning that I'm gonna come here and say capital T, small t, small t. A comma. That's all. That's what they're looking for. They give you ticks. In human, hemophilia is caused by a recessive allele of X. Of X. Anyway, it's caused by a recessive allele of X. Let's go. Of X. They're saying that in humans, hemophilia is caused by a recessive allele of X. Okay. This is carried on X chromosome, meaning that this is sex-linked. The moment you say that, that's sex-linked. Hemophilia, yes, we know is a sex-linked disorder. A woman who is heterozygous for hemophilia marries a man who is heterozygous for hemophilia. That's a woman. It means that it's like this. Capital H, small h. Yes. Marries a man who is hemophilic. Yes, meaning that the man is definitely is like that. Is use the genetic cross to show the percentage chance of couple having having a daughter who is homozygous for normal. Homozygous for normal and is a daughter, meaning that this person is gonna be like this. There's no way. Check, the father is not normal. Then it means that the answer is gonna be 0. 0. Yes, but they are saying use the genetic cross to show the percentage chance. The percentage chance is 0% because there is no way. But they are saying use the genetic cross to show. So you're saying 0% is 1 mark, but you have to show. Don't forget to show all the steps in a genetic cross. Let me see, is it P1? Let me see. A man who is, they didn't talk about anything. So I'm going to come here and say P1, F1. P1, parent, phenotype. You go to the question, you'll get the phenotypes there. I told you that this one is simple, and a man who is heterozygous, a woman who is heterozygous, meaning that this is a normal woman. Yes, marries a man who is hemophilic and a man who is affected. Then you go to genotype. Genotype, I'm just rushing so that a lady was like this because they say heterozygous, marrying a man who is like this. Yalla, let's go. They're saying like that. Don't forget that these are gametes. I always tell you that they are not at the beach, so they must be circled. If you didn't circle, it's still fine. Don't stress. Give you a tick. These are gametes. Wabona. Yeah, but how do you call this process? Meiosis. You saying P1, F1 will give you a tick. So that's the second tick. 0% is a tick, but it's a second tick. This is a third tick because we got it. So you have 4 marks. Mahala. The question is out of how many? 7. So we are searching for the 3. This is the next tick. It's the next tick. Then after that, you can draw your Punnett square here, or you can just cross. When you cross, you'll find out that this, look critically. This goes with this. So it's going to be X, capital H, X, small h. Then this goes with this, capital H, small, capital H, yes. And then this with this, small h, this with this, small h, and then y. Is there any probability of having a child who is homozygous normal and is a female? No. There is no way. Therefore, this is offsprings. F1 offsprings. And then you remember you have to cross, ne? Yeah, show cross or the Punnett square. So, and this process is called fertilization. So we give you another mark for this. But what I know that the marks are more than what we stipulate. But you saying that therefore the percentage chance is 0%. This is going to be a compulsory mark because the question is emphasizing that percentage chance. They want a percentage chance. Do you understand? Yes. So is 0% having a child who is normal, homozygous, and is a female. Yeah. Let's go to the next question. I think I'm clear. Yes. Let's go to the next question. These papers are simple if you read. If you don't read, yeah, they become sour. But I don't expect you to. Let's go to the next question. The next question. Question number 3. It means that now you have 100 marks. 100 marks. See now, you are approaching, slowly, a distinction. You're approaching a distinction. I know some of you just made small mistakes, which can be acceptable, but there are some mistakes which cannot be accepted. So, yeah. Anyway, let's go. Yeah, let's go. This is year 2023. Yeah, I know some of you gonna be successful. I pray for you so that you become successful. Anyway, learners conducted an investigation to determine the blood group was the most common in the community. I talked about a pie chart yesterday. Yeah, they conducted information about, 200. It's a big number, né? It means that the sample size. Take note of that. In each of the 3 types of blood group. Okay. Yalla, let's go. They're saying that in their committee, they did not include the first donor in the investigation. Why? Comparison of results. Okay. Yeah. The pie chart below shows, all right, let's go. The percentage of each blood group in the sample. I know you read all these questions, né? You know them. So I'm not going through too much. Né? State the aim of the investigation. Ooh. The aim must start with the word to and is always put in the first statement or second statement. That's why always I tell my students. If you can't find it, then you have to figure it out. Learners conducted an investigation to. That is the aim. From to, the investigation. The aim is to determine which blood group was most common in their community. Yes, in the learners community. Conduct an investigation. Yes. So the aim is done. Answer the following question. State 3 planning. We always ask you this, but you go back and read the question and see the possibilities for you. What are you going to plan? What are you going to do? So, but I need these people who are going to be giving me blood. So you have to organize them. Yes, get those 200 people. Yes, you are not just going to just get them. Yes, you have to get permission because this is a community thing. Get permission where you're going to do this, or the clinic, or try to go inform them so that they know and they accept. You are not going to force them. Get permission so that they can do that. You are not going to just collect the blood just like that. Get the necessary equipment, planning before you do the activity. Yeah, that's what they are looking at. Yes, something like that. State one way in which learners ensure. Yesterday I told you the word language and I emphasized it. If they ask you, don't ask you, how did this person ensure, how did the person do increase reliability, or how do you ensure reliability? They say that, how did the learner ensured? That is a past tense. Your answer must be in a past tense. The learner ensured reliability by using two. Remember, reliability is to increase the sample size by, using a big sample size by using 200 people. 200 people. You see? So, this learner used the 200 people. Sharp. So, that is done. Give the reason why give one reason why they did not include the, yes, we said comparing the result. Calculate the percentage, the number of participants that had blood group B. Show your working. Blood group B. They use 200, but only 15% blood group B. So you just come here and say 15 out of 100 times 200. So the answer you get is the answer, around 30%. Done. They give you your 3 marks. Give you a mark, give you a mark, give you a tick. Done. They're saying that name the blood group that has only recessive allele. Only recessive allele, blood group O. Remember, it is recessive allele. Remember? Yes, I said it yesterday. Results of codominance is AB. I said it yesterday. Remember, it's A and B. Give the possible genotype of the blood group represented by 25%. 25%. Yes, 25% is A. But they are saying the possible genotypes. If you remember, A is either homozygous. Go to the distinction material. You will see what I was saying yesterday. A, heterozygous. I explained this yesterday. I explained it very well. Very well. Yes. I hope you guys did not disappoint me. Ne? I don't want the marks to come back and then I'm disappointed. Say, why did I do this for my learners? These learners of mine. Ne? No. I don't want that. Ne? I want everyone to pass. Ne? Yeah. I want everyone. Everybody to pass. Anyway, let's go. Question 3.2. We are almost done. We are almost done. Anyway, question 3.2. These are dogs, man. Do you like dogs? Let me know the question in the comment below. Do you like dogs? Do you have a dog at home? Sometimes they call you a dog. And a dog. Okay. Dogs have been selected and bred over many years to produce approximately 340 different dogs breeds. They have been bred for certain characteristics desirable to humans. Saying that analysis of 786 okay, gray wolves, only direct ancestor, present day dogs bred. Okay, all the dogs belong to, I'm just reading so that I get the idea, ne? But you know, you can read it for yourself. Okay, I've got the idea, ne? Very fast. They're saying that name the process whereby the different breeds of dogs were produced. Artificial selection. Yes, these are the things we are talking about here. Where am I? Yes, artificial selection. Is it? Yes. Where is it? Okay, this is meiosis, ne? Yeah, artificial selection is they are looking for you. These dogs, even this question has these dogs we are talking about. Exactly the same dogs. Don't disappoint me. Don't disappoint me. Don't disappoint me, guys. Dogs, wolves. They're there. Born. Check. They are dogs, the wolves. That's what are the wolves we are talking about. Okay, they're saying that. Name the artist. What did we say? Is what? Artificial selection. Human. They select desirable traits. You see? That's what they said here. Desirable. They're desirable to humans. Yeah. Yes. Distinction material. Get it. Yeah. Get it. I'm talking about those who, yeah, your sisters, your brothers. Yes. Anyway, let's go. And we're saying that name the process whereby different breed of dogs were produced. Artificial selection. Describe how human carried out the process named above. Meaning that they got the characteristics they wanted. They let that individual which has that characteristic to mate with the other characteristics they want, and then the other one which doesn't have the characteristics were not allowed to give to have sex, meaning that to pass their genes to the next generation. So after many generations, they had to be with that desirable characteristic. That's what they are saying. So if you look at this example, check, it was about milk. Choose or select the cow in your herd, or now they're talking about the dogs to produce which has that characteristic. Yes. Then they are saying that let only those dogs to reproduce, the one which have the specific characteristics, then select the offspring that produce that characteristic most. Yes, let those only those offspring reproduce, allow them to have sex and have offspring like that. Keep repeating this process of selection and then the breeding until you achieve your goal. That's it, to get the desirable dog you want. Explain how it can be confirmed that all the dogs breeds belong to the same species. Same species. It means that they can interbreed to produce a fertile offspring. If they are not producing fertile offspring, then they don't belong to the same species. Yes, they must produce to produce a fertile offspring. Hence, this answer becomes right. Yes. Explain how present-day dogs may have been disadvantaged. Yes, in the relation to their common ancestor. This common ancestor wolf, Baba, it could resist diseases. Now, you have to take it to the how do they call it, the veterinary doctor. They were staying in the wild, so these dogs are susceptible to diseases. Less chance of these dogs being adapted to the environment. Yes, these dogs, the one which are new, yes, they have less chance. This one, it could adapt, Baba. Yes, meaning that some genes are being lost as you are doing this selection. Yes, so the dogs are being disadvantaged. Yes, let's go to the last question. Describe the process of speciation by geographical barrier. Guys, I told you don't go to the paper. Speciation is here. This is what they're looking exactly. This formation of new species. Exactly. There is a population of a particular species. A population of a particular species becomes split into two by geographical barrier. As a result of, you don't talk about geographical barrier. Okay, you can say river, mountain, or whatever, but geographical barrier. As a result of two parts of the population cannot interbreed, there is no gene flow, yes, between the two. The natural selection will occur independently, yes, due to different environmental conditions. As a result, the population became genotypical. You start with genotypically, then phenotypically different over time. Even if you remove you bring these two populations, you mix them together, meaning that you remove the barrier and you let them mix and they interbreed, they cannot produce a fertile offspring. They cannot produce a fertile offspring. Therefore, one or both population have become new species. This is exactly without changing any word or what. That's what you're supposed to write. That's what you are supposed to write. Someone is gonna tell me that what if I used a scenario? That's not KO because they didn't give you a scenario. KO is when the examiner sees, just give you a tick, not arguing with him or herself. Yeah, should I give no? Something like that. Don't like it. Just when the examiner sees your paper, is marking it as you are. You know, when you're marking someone, you see that someone is getting all the ticks. Yeah. You feel good, Baba. You feel good. You feel good. The same way you feel good when your marks are coming back and you have distinction. How do you feel? You feel like you are not fitting on the I win. Marks are nice. All right, let's go, man. You feel good the same way I'm feeling good when I'm teaching you. Yeah, I feel good, ne? I feel good. All right, let's go. They're saying that name the type of diagram represented above. Okay, the diagram will represent the evolution of the family hominidae. Okay, sharp. They're saying that name the diagram. I showed you these things. Phylogenetic tree, and I showed you the questions you're supposed to answer, and I don't think that they skipped those questions here. Let's go. Name the diagram shown above. Phylogenetic tree. Done. Give the letter of organisms, which is the common ancestor for all the hominids. P, because all of them they come from here. Is P. They're saying that. They're saying that. What is common? Okay, share most recent common ancestor with the gorilla. Gorilla is here. Gorilla is there, but they share a common ancestor. What shares the common ancestor is here, meaning that is this. But what shared is R, because gorilla and R they share a common ancestor. So the answer here is gonna be R. Give two organisms that have S as a ancestor. Two organisms that have S. Where is S now? S is this. Is this point. Stop it. Stop it when technology. Okay, this is S. So meaning that you're gonna say this and this. Those are the two individuals. Let's go. Name two organisms that have S as a common ancestor. We have said it. Quadrupedal. These ones. Gorillas. You know them. Is chimpanzee quadrupedal? I think so sometimes. But these ones, they are quadrupedals. Okay? They're saying that describe three anatomical features of the skeleton of a quadrupedal organisms. Skeleton, when you talk about a skeleton, you're gonna talk about the spine, the cranium, because that's a skeleton, the spine, the cranium, and then the pelvic girdle. Yes, I showed you here when we're talking about. For the case of the skeleton, here, you see, it's a skeleton they're talking about. They're saying about quadrupedal organisms. Foramen magnum is in forward position. Yes, it's a skeleton. Yeah, that. Yes. You talk about the spine is C-shaped, and then long and narrow pelvis. That's what they are looking for. Simple. Simple. As simple as eating ice cream. No bones. Yeah, let's go. Second last. Yeah. Fossil evidence is used to support out of Africa hypothesis. State out of Africa hypothesis. Modern humans originated from Africa and migrated the rest of the world. I told you when I was concluding, don't go to the paper without knowing this. Modern humans originated from Africa and migrated the rest of the world. And I showed you this. Take note of this. Unless you do not want to trust me. The modern humans originated from Africa and migrated the rest of the world. That's what they want. The word modern must be there. Modern. Don't just say humans migrated the rest from Africa. And Homo habilis, Homo naledi, Homo neanderthalensis, all those are humans. But the modern ones, Homo sapiens. Yes, they're looking for. Okay, let's go. Last question. Describe how the fossil evidence is used to support out of Africa hypothesis. How is this fossil evidence? How is this fossil evidence used to show out of Africa hypothesis, guys? How is it used? Describe how this fossil evidence is used to support out of Africa hypothesis. You guys this. I told you this and then you didn't write. Okay. You talk about all the Adipithecus, Australopithecus, Homo habilis. You say that only, the word only must be there. Only. Adipithecus, fossils of Adipithecus, fossils of Australopithecus. You can talk about the examples. Fossils of Homo habilis. Only found in Africa. Then when you go to Homo erectus, you talk about the oldest fossil of Homo erectus, distinction material, and Homo sapien. Homo erectus and Homo sapiens. Here you talk about oldest fossil of Homo erectus and Homo sapiens were found in Africa. Don't say that only found in Africa. Nope. You have to say oldest. The key point there is oldest. That's why I put it in bold italic and I was supposed to underline it so that your eyes can open when you are reading it. I think, guys, the paper was easy. Yeah. Don't forget to subscribe, like, and share. It has been M.Saidi, has been taking you all over this paper. I think, yeah, the paper is a little bit easy. It was a little bit easy. Okay, they had some challenging questions, but it was not that bad. Ne? It was not that bad. Yes. Thank you very much for being with us. Yeah. Thank you very much, guys. Ne? It has been a wonderful paper. Don't you think so? Let me know in the comment below to know how was this paper to you, based on you. How was the paper? Yeah, let me know in the question in the comment. Thank you for being with me from the beginning until guys, ne? Yeah, I just love you so much. Stay blessed, ne? Stay blessed, ne? Yeah. Maybe I just a small note that even physical science, yeah, even other papers we will be doing discussing them immediately after you have done them. Thank you very much, ne? See you.